Thank you very much for coming to the 6th Circular Strategy Symposium, Cultivating Matter. My name is Petra Gruber. I'm Department Head of Building Construction and together with Camille Breuil, the organizer of this event. I will do the moderation and introductions this morning. It is my great pleasure to ask our new rector, Dr. Ulrike Kuch, to the stage for her welcome address. Thank you for being here.
Yeah, so good morning, everybody. It's nice to see you here. Speakers, dear colleagues, dear audience, it is, of course, a great pleasure for me to welcome you on behalf of the Rectorate of the University of Applied Arts, Vienna, to the 6th Circular Strategies Symposium. I'm especially happy to welcome you here as the symposium covers some topics which I think are very relevant for the research in architecture, but also at the Angewandte in general, and also maybe for the research in architecture in general at all, in a global sense. And I think we have some speakers who stand exactly for this approach. The symposium addresses issues of global significance. Behind the question or the topic of circular strategies and cultivating matter, there lies the thread of life on Earth. I sometimes get the impression that this thread is being overlooked, especially in political circles. That is why I have the impression that I would like to emphasize it again, even with this maybe some kind of pathetic notion that it has. But I think it's important to just remember that all the time. The approach of this year's symposium to focus on cultivating matter is one that, from my perspective, and I was also educated as an architect, and I worked as an architecture theoretician. The symposium focuses on the intersection of materiality and architecture with the fundamentals of life and culture. So it's a very general and basic and important approach, in my opinion. The role of the human being and the role of architects in particular are crucial. We, in the sense as human beings, we link nature and culture. We, in the sense as architects and designers, if I may say so, we facing the challenge to moderate this process, to design material structures with the focus on harmonic coexistence of all living beings. The term cohabitation that came up a few years ago, and I think some of you have heard and maybe even written about that, this term seems to me very compelling for describing this harmonic relationship and is maybe a more rational expression for that. I understand it as a balanced ecological, social, and aesthetic being in the world. So when we look back in the history of the University of Applied Arts, Vienna, we notice that the thinking about materiality played an important role also for the thinking of what angewandt or applied means. The idea of orienting art and later design, and today even science, on the needs of a modern society around 1900, when the university was founded, is something that is still and maybe more important than ever to us. Today we want, as a university, to contribute to this harmonic, I somehow tend to use this a bit, romantic term, to a harmonic global society with art and science. To reflect about cultivating matter, about the cohabitation, about ways and strategies to implement ideas into our society, and also about the role of human beings, architects, designers, and scientists in this context is something that, in my opinion, is part of our university, and it's absolutely relevant and fitting to us today. So therefore, I would like to express my heartfelt thanks to you for coming to this university, to Vienna, to bring your knowledge and expertise into this symposium and into our university. I thank you for discussing, maybe for speculating, and if possible, I would be thankful if you also could discuss about strategies to implement your ideas into the general discourse of our society. How can we really make this happen? And I have also maybe just very shortly to apologize because I have a quite tight schedule at the moment, and therefore I have to leave afterwards. I would really have loved to stay here and listen. I know that I have the chance to listen to your talks later on, and I will do this, but yeah, I am not able to attend the full symposium. However, I would like to also thank my colleagues, especially Petra, Gruber, and Camille Breu, and also the whole Department of Building Construction for organizing this, for developing the concept of this symposium, also setting the topic, which I appreciate a lot. Thank you for that. And I hope that you all can take inspiring inputs from this symposium and bring them also to your home institutions or wherever you come from, to your maybe own worlds. And I hope you have an inspiring and a very intense symposium. So welcome and thank you very much.
Thank you very much. I'm happy to introduce you now to this year's International Symposium on Circular Strategies. This is the 6th symposium in a series that is organized by the Department of Building Construction and the Institute of Architecture at the Angewandte. Within the frame of the main theme of circularity, each year we focus on a special theme. And this year we have chosen Cultivating Matter is the Future Alive, a theme that is very close to me and my own work, as it has been the focus of my research activities for the last decades. The interest in materials has expanded from a design perspective to a perspective of global ecological importance, leading to the investigation and critical discussion of materials processing, including resource extraction and its consequences on environments. In the current situation of global warming and subsequent changes in planetary patterns, it is more important than ever to look for alternatives to our current technologies, pedagogies, politics, and responsibilities. As a building construction department, we are challenged to support a paradigm shift from static, everlasting building towards dynamic, life-hosting processes. To stress this urgency, I would like to cite a recent publication of the Potsdam Institute for Climate Impact Research of last week, 12th of October. It's called The Road Back to Paris, and it's authored by Rockström and others. And it says, a pathway to stay below global warming temperatures increase of 1.5 degrees Celsius would require halving global greenhouse gas emissions by around 2035, halving the emissions, reaching net zero CO2 by mid-century. That would be the consequences. Every 1/10 of a degree of additional warming increases the risk of crossing tipping points in the Earth system, including ice sheet loss, permafrost thaw, or weakening ocean circulation, as it's already happening. Returning to below 1.5 will depend not only on decarbonization, but also on protecting and restoring natural carbon sinks and other life-supporting functions of the Earth system. This includes transforming global food systems, stopping deforestation, and keeping freshwater use, land system change, biodiversity loss, and other key indicators within planetary boundaries. And I think this really stresses the importance of the topic and how we as architects and people working in this area can contribute. Cultivating is our choice of activity, as it encompasses the ideas of culture as something inherently, but some argue not exclusively, human, and cultivation, the purposeful care to propagate and grow organisms and ecosystems. Since millennia, humans have elaborated ways to create what we like to call cultural landscapes that leverage and foster natural rhythms. The adoption of cultivation and care to our architecture and building activities requires learning and experimenting on all levels of scale. Technology is what brought us to the present situation, and it is clear that however we continue, we are part of a global biosphere that, according to the advocates of the Gaia hypothesis, is a whole living planet. Biology is the only other model we have to reshape technology into another direction, and for architecture, that is living architecture. Learning from and integrating living organisms into our buildings is the goal of this approach. We hope that the learnings from the event will help us to envision a future of more sustainable materiality, but also more livable futures on this planet. The symposium is structured over 2 days, featuring 2 keynote lectures, one by Carole Collet and the other one by Mitchell Torkim, who is hopefully just arriving in Vienna, followed by sessions discussing relevant topics such as design with organisms, bio-inspiration as an approach, artistic approaches, prototyping and fabrication, bioreceptivity, and critical reflection. In the afternoons, we will host a poster session and present the exhibition of the research project Root Arc in the atrium space. During the breaks, you're welcome to browse and also find prototypes of speakers exhibiting in the atrium. After each session speaker, we have a short transition time for 1 or 2 urgent questions, and at the end of each session, we will have time for a discussion that is moderated. Please be aware that we do record the event on video and take photographs. The lectures will be published on the YouTube channel of the Institute of Architecture, and the book of abstracts is already available on the website as a PDF download. And you also find the QR code that leads you to that posted around this space. Before I introduce our keynote speaker, Carole, I would like to introduce you to the team behind the organization of the event. Camille Breuil is the main curator and of the symposium from concept to every detail. The other members of the Department of Building Construction and Institute of Architecture helping are Anja Jonkans, Andreas Körner, Michaela Karpov. Where is she? Okay. She will be running around probably. Claudia Rüssli. And I want to extend my heartfelt thanks to this wonderful team organizing everything. I also want to thank the technicians of the Angewandte for running the show here at this moment. And I want to thank our moderators who will guide us through the symposium days, today and tomorrow. I want to briefly introduce here Anna Orbanić, Academy of Fine Arts, Vienna, Helga Lichtenegger from BOKU, Ebba Franzen-Waldöhr from Angewandte. The poster session will be moderated by Andreas. The keynote tomorrow will be moderated by Anja. Victoria Sandor will be one of the session chairs tomorrow from the AIT. Is she here already? Victoria? Not yet, probably tomorrow. Barbara Imhof from the University of Innsbruck and Liquifer Systems Group. And Gonzalo Vallo, I hope I pronounced this all right, from the University of Innsbruck. The closing words tomorrow will be held by Camille, and she will also introduce to the Root Arc exhibition. We are very grateful to have the support of the Rector of the Angewandte, Dr. Ulrike Kuch, the former head of the Institute of Architecture, Bärbel Müller, and the current head of the Institute, Sam Jacob, for the continuous support of the theme of circular strategies and this yearly event. Thank you also for the many other people from the Angewandte and beyond who are and were involved and are available for discussion and information along the way. A very special welcome I want to extend to the students. You are the ones who will carry these ideas further into the future. And with this, I want to introduce our first speaker today, Professor Carole Collet from the University of Central St. Martin's in London, UK. And her lecture is called Renewing Nature, Enacting Biodesign to Restore Ecosystems. And the lecture will situate how the practice of biodesign can stimulate new ways of knowledge to inform how we design in the context of our planetary boundaries. With a focus on disruptive biodesign research, Carole Collet founded the Living Systems Lab at Central St. Martin's in 2013 already as a pioneering research group to explore living system thinking for the pursuit of new ecological knowledge in the creative sector. She curated the first international exhibition that situated biodesign via the lens of sustainability, also in 2013. It was called Alive, New Design Frontiers, and was held at EDF Foundation. And she published an original framework for designing with the living. Her design work has been featured in international exhibitions such as the Vienna, Saint-Étienne Design Biennale, and the Pompidou Center. She's also director of Maison Zero, a platform for regenerative luxury co-developed with the group LVMH to leverage creativity at the service of ecosystem regeneration. And she will also introduce us to this. She is recognized for her leadership in ecological design centered on biodesign and biomimicry principles. She works as a design researcher, curator, and educator. She has forged the integration of ecological values in the design curriculum over 20 years with the creation of radical new master courses in textile futures, biodesign, and MA regenerative design. She regularly contributes to international conferences and publications on the subject of living systems design, biodesign, and regenerative futures. She was awarded a Design for Planet Fellowship at the Design Council of the UK in 2022. So with this, I thank you very much for being here today. I wish you wonderful 2 days inspiring times, and I would like to ask Carole to the stage. Thank you so much for being here.
Great. Thank you very much. I'm delighted to be here with you today. I'm really looking forward to the excellent program you've put together. It's a bit of an indulgence for me, so I'm grateful to be here. A warm thank you to Petra and Camille for inviting me to share some thoughts with you today and for looking after me so well. We have 45 minutes for me to share some thoughts with you, then we'll have 15 minutes for questions. And when I was preparing this, I felt I had two options. Either I would take you through an in-depth analysis and presentation of some of my research work, or I could showcase a breadth of approaches. And I decided to go for the latter, because I think it's really important that we look at a diversity of strategies, of design strategies informed by circularity, living system thinking, because we are faced, and Petra announced it really well in the introduction, we're really faced with quite a dramatic environmental set of challenges, and we're confronted with what I call a wicked problem, and there's not one way to address that level of challenge. We need to really look at it from different lenses. So I'll introduce myself a little bit more. When I founded this research group, the Living Systems Lab, in 2013, it was still very early on. And you will see what I will be showcasing today is beyond the diversity of projects, it's very much a journey. Some of you are at the beginning of this journey, some of you are halfway through, some of you maybe towards the end of this research. But for me, it's really important to showcase how in a short span of time, things have changed a lot. In the year 2000, we talked a lot about living technologies. This was really the onset of synthetic biology. We knew a lot already about the climate crisis, about the biodiversity crisis, but it felt that ecology and the way to think about how living systems work was really not the central space to explore new design ideas. So I set up this lab to really look at how can we learn from living systems, learn about ecological principles to inform creative practice in a way that we're not just using nature, but also designing for nature. This leads me to a whole set of activities which are interconnected. And what I really like, we discussed it with Petra earlier, I'm a maker. I'm a textile designer by training. And you will wonder why textiles in an architectural conference, but actually textiles, we are at the heart of the materiality of our everyday. Whether we design textiles for a fashion application, for an interior or architectural application. And for me, as a maker, it's important to evolve and develop new ways of thinking, new ways of making. However, I think it's also important to really look at how we situate our practice and how we can frame new knowledge. So I navigate a whole range of activities between curation to making knowledge to writing up, analyzing the work, all the way to using this research to inform the development of new design curriculums and developing new courses, in this case 3 master courses at Central St. Martin's. We also have, as part of this research group, a group of PhD students, and I will showcase some of their ideas today. So I will intersect some of my practice and some of their practice. That Living Systems Lab is really where I will conduct more of my fundamental curiosity-driven research. But then I have another role at Central St. Martin's. I'm the director of Maison Zero. Maison Zero was set up in 2017. It's the name of the partnership between the luxury group LVMH and Central St. Martin's. LVMH, you may know them better for their champagne or for their couture houses. They actually employ 1,000 architects. So you may not know that, but actually we work a lot with architecture in relation to that practice, whether it's for designing factories of the future or designing stores of the future. And at Maison Zero, we also are very much centered on this notion of rethinking how we can design, but to go beyond circularity. How can we go into a space where the act of design contributes to regenerate ecosystem? And we do that via a range of activities across scholarships, awards. We have a challenge fund for some of our courses, research projects, as well as developing new courses. So we validated the new Masters of Regenerative Design as part of Maison Zero. And this gives me another, another role, another place to play, because this is directly working in relation to the constraints of the real economics of working with companies. So I work with different brands at Maison Zero, and we're really looking at the concrete pragmatic realities. If you are a large-scale company transitioning to circularity, which is a core objective for LVMH, one of their key strategic environmental objective is to reach 100% eco-design products by 2030, and this is all based on circularity. But you work within a constraints of commercial context. So I've got both heads working together, the very playful fundamental research with living systems, but also the academic rigor. And then I have the much more applied knowledge that we develop through Maison Zero. So I feel in a very privileged position because actually I can work across a whole spectrum. When I talk about a living systems approach to design, quite often people say, well, what do you mean? What is it? How does that work? What are you doing? And it's very much really looking at what we can learn from understanding nature better. Sadly, I feel that as designers, and still in the mainstream design education, whether it's in design for product design, architecture, fashion, we don't place ecology at the heart of the curriculum. Yet, as designers, our activities have a radical impact on ecosystems. Coming from textile design, I studied textiles about 30 years ago now, and that's when I understood the dramatic impact of my industry on the environment, particularly in relation to water usage, chemical usage for dyeing, for printing, the forever chemicals to waterproof clothes. And I, straight away, 30 years ago, I just decided I don't want to be part of the textile industry unless I can reconcile ecology and being a creative textile designer. But I was never taught about ecology formally. This happened simply through a lot of my research and reading. But what we don't teach and what we don't know quite often is when you look at an ecosystem, so I chose one here, what you see here is not just a few trees and a path in a forest. Of course, this is a moment in time, but what you see here is a whole set of visible and invisible, dynamic, interconnected relationships. What's going on in the soil, a multitude of dynamic interactions with the decomposer organisms, the fungi, the mycelium that will exchange nutrient across root systems. You have, in this case, leaves falling and preparing nutrient for the next season. You have a lot of interactions that you don't see or don't hear, which are there. And as designers, we often create the conditions to disrupt and destroy such an ecosystem. So if you're a furniture designer, you want some wood, you're going to say, okay, we need a walnut wood. You think about it as a material. So we have that very extractive approach, without thinking that cutting that very tree will destroy the whole habitat of a multitude of species. Some will not recover, some might be able to recover from the act of cutting that tree. And I reference here Petra, who really talks a lot about this system thinking in terms of biology, and who says all living beings are members of ecological communities. They are bound together in a network of high interdependencies. That's fundamental to design. This should be at the core of what we do. So we need to understand the consequences of our design decisions and how they disrupt these networks of interdependencies. And this living systems approach is very much centered on that understanding that we can nurture, support, or disrupt a living system, and this will have a massive impact on biodiversity and climate. Our current worldview, if you look at the mainstream worldview around the world, is very anthropocentric. So we look at nature as a resource. We need it for, concrete. We need some sand. We need wood for architecture. We need glass again. We need some sand. Or we look at nature as a waste ground. There was a pictures recently showed of a desert in Chile where you can see a massive landfill from space of fast fashion waste. We never see later, and you referenced, Gaia, the planet is a living organism. Lovelock positioned this very early on, but we tend to really accept nature as a resource or waste ground. We need to shift to this next worldview, which is much more Earth-centric, so that we are only one species amongst many others, and we need to look at how we, of course, design for us humans in a way that actually supports circular strategies that we find naturally in ecosystems. That also requires thinking about shifting our language, and Kimra talks very well about this when she says if a maple tree is an it, when she's referencing the English language here, if a maple tree is an it, we can take a chainsaw. If a maple tree is a her, we might think twice. So we need to really start looking at setting these different relationships with the natural world. That means we need to move from a system which is extractive, degenerative. We're still very much in a linear economy, even in architecture. It's very toxic. We're generating a lot of toxic materials. We found, there was a report last week, in the UK, all rivers but one showed evidence of forever chemicals. All rivers in the UK are profoundly impacted by microplastics. Microplastics and forever chemicals are fundamentally linked to my industry, the textile industry. This is the way we work. We need to shift this to a way that we design, thinking in relation to multi-species kinship, to design to nourish ecosystem, regenerate, so shifting to fully circular sets of economics, and design in a way that is life conducive, carbon capture, inclusive. Of course, we need to think about the social dimension, and much more holistic. So this doesn't require a step change. This requires a systems change. And that's really important in terms of setting the scene to what I'm going to show you, because some of my work is quite looking at radical shift, radical new ways of thinking, which are not possible yet. And others are really looking at what are the little steps we can do to really accelerate that transition to circular regenerative mindset. Everything for me comes from learning from how nature works, and also about being very humble. As humans, We think we're very sophisticated. We definitely, we're now all the way going to Mars. But actually, there are very simple tricks in biology we still don't know how to do ourselves. And I think it's important we really pause for a moment and think about, well, how does nature truly works? And this was really and how the practice of biodesign emerged, really looking at what we can learn from biology. So here is a leaf from a tree that grew in a matter of a couple of days at springtime, developed completely the architecture of that leaf systems, the color, but also the capacity, the function. This is a photosynthetic system that can capture sunlight, turn it into sugar, eating carbon, and releasing oxygen, which we breathe. All of this highly complex system done and produced and fabricated at ambient temperature in a matter of few days using only local resources. Local because the nutrients came from the roots of that tree. The sun system, the solar system is round, the water system is the rain. So all of this was done without producing any byproduct that brings any toxicity in the system. And this relies on a principle which is extraordinary. And to this day, in my view, still in the realm of the magical for me, is this notion of self-assembly. So if you think of a cell, you are the result of self-assembly. You were, a long time ago, one cell that multiplied into 2, into 4. And this self-assembly, and I have to say, even in science, what is life and how life works is still a very central question. We don't know how this cell differentiation, cell multiplication, means that one cell ends up being your hair, one cell ends up being your eye. It's still a very complex system. And that self-assembly principle is core to synthetic biology and has led to really innovative new generation of materials, which I'll mention a little bit later. At that cellular level, this is the same leaf we saw in a previous picture. This is a Sampolia, which is a plant you find that originates from South Africa. But you can look at this. This is, again, grown at ambient temperature. The membrane allows for a gas exchange, so it can actually capture carbon but release oxygen. Every cell is designed to function, to breathe, and to capture sunlight. This has grown in a matter of a few days at ambient temperature. We don't know how to do this yet. And for me, a turning point in my career was, and I remember very well, Janine Benyus, 1997. For me, this was a revelation. I had been looking at how can we really design and make textiles in a way that's not as impactful on the environment. And after Janine Benyus, I just thought, okay, this is it. The answers are in biology, in how nature works. And she says life can't put its factory on the edge of town. It has to live where it works. As a result, nature's first trick of the trade is that nature manufactures its materials under life-friendly conditions, in water, at room temperature, without harsh chemicals or high pressures. We still don't know how to do this when we make a textile. So we think we are very intelligent species, but as said, actually what we produce is fairly basic compared to how nature makes. So this became a central research for me, that all my research inquiries are driven by how can we get a step closer to how nature works, but also how can we design in a way that restores and regenerates and nourishes ecosystem. So very early on, a central research question became, how does nature make a textile? Not a fiber, because when you're a textile designer, if you think of a cotton fabric, you will harvest the cotton fiber and then turn it into a fabric. But I wanted to go a step further. If you look at, if I want this systems change, I don't want to tweak what goes on in the industry. I want to look at, could we grow a fabric? And actually, nature does it. And this is what we call a lace bark. It comes from a lageto tree. It's a tree you find in Jamaica, in particular. It's the inner part of the bark of the tree. And you can see it looks like a woven cloth. If you look a little bit closer, you can see all these interconnected. So if some of you have ever looked at some of your own clothes, if you look up straight, this is what your own fabric could look like. This has grown slowly, in this case, at ambient temperature, is a tree. So there's a code in nature that can lead to the fabrication of a textile.
So for me, that became straight away, where it's interesting. So there's, the genetic information exists in terms of fabricating this. Another example, this for me looks a little bit more like a 3D printed object. The first time I saw one, I thought it was 3D printed actually, because I only saw one bit of it. This is a sea sponge. So the top bit is quite a hard material. And you can see at the bottom, so this is a sea sponge that would be anchored. So this would be the part anchored in the sand at the bottom of the ocean. And you can see the little hair on the side. So that hair is actually quite brittle. But aside from that, it showcased, and there's been some scientific publications around that, it has superior quality compared to a fiber optic made by us humans. So this is the way nature makes a fiber optic. But it's of better quality than the ones we make. And when the ones we make is following a system of, we take from nature, we extract materials, we make them in a factory with high energy, and then we don't design them to be recycled or to become a nutrient again. So we have that linear approach to making. This is grown at ambient temperature, superior quality in terms of fiber optics. So I feel we have a lot to learn from how nature works. And what I want to share with you in the next half an hour is really looking at how I use this knowledge and this inquiry and this research to try and get a step closer, and it's very difficult. So on the one hand, I work with much more what we call curiosity-driven fundamental research. This is very much the Living Systems Lab, all the way to applied research, because I actually work with companies and I also do contract research with companies. And I navigate three kind of methodologies, from speculative design to biodesign and regenerative design. Speculative design was something I was doing much more 15 years ago when I was beginning to look at biology, because biodesign didn't quite exist yet, and we were looking at how we can do it. We hadn't set up, we've set up our grow lab at Central St. Martin's in 2019 now, so we didn't have access to biology labs. But I wanted to look at the imaginary of what could be a way to use biological system to rethink radically a textile system. And the other thing I do is I work across species and ecosystems, and I will come more evident as I go through the work. And the other aspect of work, and this is something we discussed with Petra, for me it's really important that, yes, to use our creativity and our creative inquiries to lead to new knowledge, to new research, and to situate our practice. But for me, it's important to really understand how we situate that practice. And when I started working across biodesign, actually, one of the first things I did was actually curate an exhibition looking at what's going on in this space. The exhibition that Petra mentioned, called This Is Alive, was in Paris. And at the time, there were a lot of practitioners across architecture, art, design, talking about biodesign. But I could see a whole range of ideas there that were quite disconnected. And for me, I just felt like we need to really have a consistent, rigorous understanding of how can biodesign practice lead to systems change that will address some of our environmental issues. So I developed this framework looking into when a design is simply bio-informed. So you might learn by, and then we saw a lot in references in the early 90s of biomimicry, looking at a bullet train that's based on the beak of a bird, or a car designed following the aerodynamic form of a fish. But this was sort of mimicking a form or behavior or function, but still manufacturing in the way we do, with human manufacturing, high energy extractive materials. However, some of this bio-informed practice leads to really looking at how ecology works, how ecosystem works, how behaviors work. So it's a very, very important place in biodesign. Then we have all the biodesign practice, which is bio-based, so actually using materials like bio-waste, so agricultural waste, for instance, and then transforming them. And then we have what I call the bio-integrated. So this is when actually you design together with an organism. So you integrate their biological capacity to control the morphogenesis of a system or a shape, and evolve it together with an organism. So you sort of co-design with an organism, you create the relevant environment for that organism to produce something with you, for instance, a mycelium material. And then the bioengineer, this is when you actually go into genetic programmation or genomic programmation. And each of these categories have different ethical implications and different contribution in terms of sustainability. And I work across all of these, and I think it's important for me to understand what does it do and where do I situate my practice. And the other form of design I practice is regenerative design. And regenerative design is ecocentric and biocentric in that we need to understand, again, living system thinking and how we can adopt that in our design decision process. But regenerative design is a place-based practice where you choose an ecosystem, you work with an ecosystem, you understand its complexity, what is off balance. Most ecosystems are off balance now. Understand the challenges, the issues. You look at the local climate conditions, and you look at the need of the communities or the cultures, and you look at combining these three different aspects. And the product that you design, or the system or the service you design, is a consequence of the restoration of that ecosystem. And I will showcase examples across this as well. I want to start with talking about a few projects which have evolved from speculation to actually biodesign. And again, 15 years ago, it was very difficult to access a biology lab, and this is how I began that process. I have an obsession with plant roots. I saw that you have Diana Scherer's work here. I'm a big fan. I love the edge of a plant. So plant hair and plant roots. I don't know why. This is what I'm obsessed about. And 15 years ago, I won't show this project here, but I was working on a project where I was paired with a Nobel laureate. So very humbling exercise. I'm a designer in conversation with Sir John Sulston, who got a Nobel Prize for his research in cellular apoptosis, so how cells commit suicide to allow for other cells to develop. And part of the project was these conversations and discussions, and I was telling him I'm really excited about biology because I'm looking at finding better ways of making, fabricating, cultivating matter. And he said, oh, well, if you're interested in this, you need to look at what's happening in synthetic biology. And this was 2007. I'd never heard of synthetic biology. And I was like, what is this? Started reading a lot of scientific papers, looking at what was going on. 2010, the first cell, so-called, it's controversial, but the first so-called synthetic cell called Cynthia, produced by Craig Venter, was announced. And this was the first time that an organism on Earth was designed by a human, designed on a computer, printed the DNA, booted the DNA in a cell, and that created a whole new organism. And at the time, we were looking at a lot at this disruptive innovation in biology. There was a lot of research on the sort of synthetic biology roadmap in the UK, which was published. And everybody was looking at the use of synthetic biology to rethink our medicine, so to input on medical research, or the future of oil. So can we grow oil systems? And I was the only designers in these scientific meetings saying, no, but actually textiles should be an interesting place here, because actually there's a lot of things we can do in terms of coloration, in terms of finishes, a lot of the toxic finishes we use on fabric. But at the time, there wasn't really an interest in looking at textiles. But for me, it was critical to think, okay, where could we go? Opening up the imaginary of that technology in a textile context. And I wanted to look in particular at a future textile production, looking at a 2050 horizon, based on root systems, because I'm really fascinated by them. And some plants have extremely long roots. So for instance, this is a rye plant. The surface can reach about 639 square meters, but the underground root system can be 130 times greater than its surface. So there's a massive hidden capacity underground there, which I thought was quite exciting. You also have an incredible amount, and these are only a few of them, there's a whole range of different root architecture, different strategies to pump water, to use nutrients, to exchange nutrients, are used by different plant species, from a very central root system to a very decentralized, deployed root system. This always adds to the resilience or not of a particular plant species. But this plant architecture, root architecture was exciting for me. So I decided to explore what could be the future of this synthetic biology. We talked about living technology at the time, through this project called BioLace. And BioLace was featured in 2050 at an environmental crisis time where growing food became problematic. and where we were moving our agricultural system to vertical food factories. They exist today. They didn't really 15 years ago. And I really wanted to look at, if we're looking at a climate horizon of 4 degrees plus, we've missed the 1.5. This was the news this week as well. It will be much harder. How do you grow food when it's 50 degrees Celsius outside? A lot of our farmers are already really struggling to control crop growing. So I wanted to look at this sort of new potential, and I decided to create this project called BioLace. It's a whole series of combination of photography and also hands-on practice to look at the future of an agricultural system where you can produce food and fabric at the same time in vertical hydroponic factories. So strawberry noir is a black strawberry which is enhanced with level of vitamin E because we need more vitamin and antioxidants, and the black lace is for fashion trimming. Basil number 5. This was really looking at developing a basil, a herb for culinary use, together with a scented perfume lace. Factor 50 tomato. This is all based on some scientific research. So in tomato, there's a chemical called lycopene, which actually is useful for us to in terms of our skin, in terms of sun exposure. So this plant was designed to produce bacon flavored roots together with tomato. This is the famous BLT sandwich in England, the bacon lettuce sandwich. But the tomato were enhanced with lycopene. So actually, by eating them, you would get a sun factor protection. And then the gold nanospillage, this is also based on research on biomining. Some plant systems can extract minerals from soil and from earth. At the time, I was looking at a lot of eucalyptus found in Australia that were actually gold mining. So this is a gold nano spinach. Again, it's a form of spinach, it's edible, but actually can recover particles of gold and bioremediate. I also, at the same time, try to work with real roots. So this is an actually lace doily. It's quite small. It's about 12 centimeters, made from real strawberry roots. And I wanted to really look at exploring, could root systems be flexible enough to enable a construction of such a complex lace pattern? But also looking at this proposition, so the idea of harvesting food and fabric at the same time. And this project was really interesting because I worked a lot with the textile industry. I was showcasing this as a provocation in the textile industry to make them realize that the industry, at the time, it wasn't really acknowledged. There's a lot of change has happened in 15 years. But a lot of people were reluctant to even look at environmental strategies in the textile industries. And I wanted to provoke them to look at, you depend on nature. In 2050, if we miss our climate target, it will be difficult to grow cotton. You won't have cashmere, you won't have wool. How will we grow fabrics? So this was a very useful tool to use to, in a way, shake the industry. But then I moved into, could I actually do this? So working in biodesign practice, looking into actually, can I control root systems to create a pattern? I'm really interested in patterning. This is my foremost background. So I developed this project, Geotropic Textiles. This was showcased at the Pompidou Center, looking at 2 key questions. So one was, can I create a loop system? And if you remember the Lagetta laghetto tree, the lace bark we saw, this was really constructed, through the way the tree grew. So could I control a lace system to create these loops? Creating loops is fundamental if you want to do a knitted fabric, for instance. And the other question was, if I was to seed the plants with a pattern, could the roots echo that pattern in some way when I control this? And I worked with tropism, phototropism, graphtropism, so the way a plant responds to light to grow up front, whereas the roots will grow down. And I developed a range of projects. I won't show you the whole project, it's very complex, across different plant species with different types of root systems. And I'm going to share with you, so this is some of, of course, documenting the research, all the lab sheets. And I'm going to show you, so this was the most successful one. This is a mustard seed, very fine root, slow system. And through phototropism and gravitropism and controlling the light environment of these plants, I managed to control creating loop system or these sort of basic woven structures. And this one was the complete failure. I like to share failures because I think it's important. We develop new knowledge when things don't work. This is a flax, this is a linen. And this one was fascinating. It actually was a headache, to be fair. This was really looking at, so the closer the seeds together, the more chaotic it became. And we do know that plant species communicate, and some plants do not like a neighbor, even if it's from their species. Others welcome it. And in this case, flax did not want. Every time the seeds were very close together, it became chaotic. And the further apart, the more control I had. But I never managed to control anything. So partly because the system grew very fast, but the roots were not growing linear. They actually were going in all different dimensions. So this one didn't work. But what the knowledge established there was that actually we can control root systems through phototropic response. In most of the plant species I tried, but in some of them it doesn't work in terms of controlling a pattern. The other obsession I have, so we've seen the roots, is fur. So again, that very curiosity-driven approach. Plants make fur. And sometimes, so on the right, you can see this is more cotton grass. That would be North American. You'd find them in prairies. And in this case, the fur is actually the seeds. So this is a strategy to disperse and disseminate seeds. Same with the grass. But on the right, it's a cactus. And that's for sun protection. So it's grown a fur to protect itself from extreme sunlight and extreme temperature. If you look at the plant world through their fur, you'll see a whole different world. And sometimes, all plants actually have fur on their stems and their leaves. Most of the time, they're not visible. And in this case, they're extremely visible. But I found this space really interesting. And I wanted to look at, well, what does this mean for textiles? Could we actually really look at this? And again, looking at what does this mean? And at a time, there was a lot of discussion around, a lot of critique around the ethics of living technology. And some research going into looking into, and a quote here from Juan Henriquez in his book published in 2001, saying that, now that we have this synthetic biology capacity, we can now read and rewrite the gene and actually intersect bacteria, plants, and animals. We can actually cross the divide between our typology of animal, insects, and plants. And what does that mean? So I am an anti-fur designer. I do not value the act of actually killing an animal for its fur. So I wanted to look at, could this be that we start looking at crossing this divide for the benefit of, designing this new very warm material, fur? It can be useful in some ways. So looking at a fungi that can make fur, or a plant. This is the alocasia that can make a fur. And really looking at what would that mean to reach that stage and reach that level. And then, same as with BioLace, I took it into practice, looking into what are the plasticity. Can you actually work with a plant fur? And you will find actually there's now 2 companies set up. One makes a filling for down, for like winter jackets, made from a plant grown in regenerative system, and it's the fur system of particularly of the flower stem. And then there's another plant that's working with plant fur as well. So this is concrete now. This has evolved. And I was looking at the pace at which, so looking at nature's rhythm is another dimension that we need to look into, because the cotton grass, for instance, you saw earlier, fast growing, grows really quickly. But if you look at, this is a cactus. Cactus grows on average 1 centimeter per year. This actually means that these 0.28 gram of fur took 9 years to grow. So we need to also understand nature's rhythm. Some plants are very fast at producing matter and cultivate matter. Others are extremely slow. In this case, I developed a collection of miniature textiles to exaggerate and showcase this scarcity and this slowness and the preciousness of this material. So what I do, I tend to intersect working across this curiosity-driven, is driven by a practice, together with biotechnology, biocraft. So I really operate around industrial application. It's scientifically informed, but I also bring in this artistic and craft-based approach. And that leads me to work with biotechnology textile companies. So recently I worked with a company based in Germany called Amsilk. in that, I can't talk about these projects, they're confidential, but what I can say that my role is I'm a designer in there looking at a sustainable approach of the material we're developing. And same with Epoch. Epoch is a new startup based in the UK, developing a bioenzymatic nylon, and we're currently collaborating on a project together. They'll be in a public domain at a later stage, so hopefully you can see them later. But we have a lot of work with industry, and based on this sort of curiosity-driven research, we have companies coming to us interested in what does this mean for us. And we've just worked with Colorifix. Colorifix is a UK-based microbial dye research company. They've developed a system to develop microbial dyes that are really reducing the impact, and you can see on the right, in terms of the environmental impact compared to the synthetic dyes. Synthetic dyes are very toxic for the environment. And they are funding a PhD research to look at how we can move from dye to print. So printing textiles and dyeing textiles is actually two different, quite different processes. So this PhD is looking at actually how we can adapt this dye microbial system to a print system. You need a print paste, for instance. What are the best print pastes for these microbial systems? And she's also developing, this is Rose Lloyd, developing a color system that the color factories can adopt so that they know to get that pink on a polyester, you need these two colors, because it's actually quite counterintuitive. And so she's developed a whole range of tests and working across the two colors which are currently established as working in terms of color fastness and light fastness, are the blue and the pink. With Maison Zero, we did another research looking at much more the high-end luxury potential use of microbial dyes. And this is Charlotte Verse, who was a designer in residence with us, looking at using, so this is a natural microbe that will express this purple dye. And she designed a machine to print with a microbial. So you can see here the nutrients are in the bottles, the fabric is fed through, and at the bottom of each truss, this is where you would have the liquid microbial dyes. And depending on the pace at which you circulate the fabric, if you fold it before, if you pleat it before, you get a whole range of different one-off unique patterns, but yet repeatable in terms of the systems. So it's looking at much more an extreme automation of printing and dyeing with microbial dyes. We have a similar research in terms of circularity. This is Heijo looking into carbon capture, biomineralization processes for future product design. She's established a whole library made from waste, across ceramic waste to paper waste to polyester waste, and looking at how we can actually use these waste, biomineralize them for future product design application. So this was really looking at how we intersect curiosity-driven with a little bit more industry-facing and scientifically facing research. Moving to the world of fungi, again, and I won't spend too long on this, but for me it was very interesting to look at what can we do in terms of fungi. At the time when I started that research, there was a lot of mycelium material research, but I was again interested in the patterning potential of mycelium on textiles. So could we actually achieve things like, we talk about tie-dye in textiles, so this is a tie grow. And this controls the patterns because you simply tie as elements of your fabric, you let the mycelium grow, and you get this tie-dye effect, which is also hydroponic where the mycelium rests. Looking at using mycelium to create permanent pleats. So a permanent pleat in textiles can only be achieved with a polyester. If you pleat a cotton or a linen, when you wash it, the pleat will disintegrate, whereas the polyester is heat set. I was looking at could we use the mycelium that will actually partly eat away some of the fabric, and when it's in position, actually it changes the architecture and the structure of the material at the point of the pleat, so that actually when you bring it back, it actually becomes a permanent pleat. So a lot of research into that, looking into embroidery system, looking into what we call the lace assembly. This is all made from off-cuff leftover. Can you recompose, assemble textile constructs working across mycelium? And this one I wanted to share again. This one is a failure and still a strange phenomenon that I haven't managed to replicate. I've tried 3 times now, it hasn't worked. This is at a time, and we were talking about this with Petra, so before we had a grow lab at Central St. Martin's, I had my own DIY lab set up in my home studio. And my very first encounter with mycelium, of course, you set up your experiments, you try, And this is actually, I'll show you. So this was coffee, Mycelium loves coffee, which I had compacted in a petri dish, and it was inoculated with a culture. I won't tell you which one. And I left it, and at the same time, a lot of other cultures. It was sealed in a bag, a container, and all the others worked. I could see the material growing, and this one, I was looking at. If you look at the right-hand side, I could see it wasn't complete. It was just like there was still some patches that were still, the coffee wasn't eaten. It was quite hard to see in the bag, so I just thought eventually it's not working, I'm getting it out. So I got it out, and then I thought, oh, this is weird. So there's contamination, of course, you can see. But then the material felt really quite rubbery, and I thought, this is strange. It's done this quite strong material, but it hasn't grown. And so I baked it, so I killed the mycelium, and then I washed it. And then I discovered this. I was like, what's going on here? So I'm a textile designer. In textile design, you often are asked to design floral patterns, and which I always avoided. And then here I am, opening this bag and finding that my mycelium has designed a set of floral patterns. They are actually more fractal, as you can see. I've tried to replicate that experiment with the same culture, the same setup several times. It hasn't worked. And what has happened here is actually the contamination. I don't know what contaminated it, but that organism resisted the mycelium. There was a battle there. That meant the mycelium couldn't go around and created that fractal response. And this is, for me, why I love working in biodesign, in biology, because this is the power of biology. Not one that I could control in this case. I'm still trying. But I think it's really important to leave room for these experiments. And actually in science, quite often you'll find biologists will discover something by accident. And this was a true accident. And then I carried on a little bit more research. Recently, this project is not complete, but I wanted to look at what happens in terms of, there's a lot of mycelium material development now, particularly in terms of leather, used in architectural constructions too. But I was interested in what mycelium use can do to rethink a craft practice. So I was looking at when you kind of work across materials, in this case wood, and reeds, and how can you actually develop a whole new craft typology working across mycelium technique with two particular objectives. So one was to augment the material surface. So this is bits of wood, and you can grow it so a little bit like a reinforced concrete structure using something to hold it together, grow the mycelium in between, you double the surface of your material. It was actually quite difficult to control, and I had to bring this Japanese technique of actually burning the wood to control more or less where I wanted the mycelium to grow. But the other objective for me was to look at, could I create an inlay? I'm really interested in wood inlay, but I wanted to look at, could you create that inlay effect with mycelium? And doing a lot of different sampling activities. So in here, the wood inlay worked really well, but I looked at the three-dimensional composition. So this is really exploring the language, the typology of working across materials with mycelium systems. I don't work with automation. I'm a craft maker. For me, it's working with the handmade that really is of interest to me. And then I developed a whole range of products which were showcased at the Saint-Étienne Biennale this year, and looking into how we can actually really use this new way of controlling basketry, controlling mycelium for basketry and woodwork. And I'll conclude with this last project. You've looked at how I worked with particular species, but I'm also interested in working with ecosystems. So an example of a PhD project, which is looking at that system thinking. This is Cassandra Quinn, project funded by Monsoon Accessorize. She's looking at the flax linen system, and again, quite a complex system, but when you cut your flax plant, you need to separate the stem and the linen fragment from the rest of the plant. And this can be done in a closed tank. It's called tank retting or water retting. But actually the water of that tank retting, so it's actually, if you're looking at a lot of linen is produced in Europe, Masters of Linen, the certification for Europe, will not allow tank retting, because if you clear the water, it's so rich in nutrient, it actually is problematic in terms of pollution for rivers in particular. So she's looking at bioremediation with algae for this tank retting system. Because currently the tank retting is done, you cut the flax, you leave it on the field, and it's the dew in the morning that would slowly disintegrate the flax, so you recover the linen. But with climate change, that dew rating is becoming problematic. So she's looking at using algae to remediate this too rich wastewater. But out of this, these algae are then used to develop textile pigment for printing and dyeing. So it's about looking at that system level. Another PhD looking at Rome, so looking at what's going to happen with climate change in Rome. And actually some of the practices, some of the smell, the sense of Rome will disappear because of climate change. So she's looking at what are these cultural heritage connected with a very Italian culture, where the scents will disappear. And she's recreating this scents using fermentation and biodesign practice, so that in 20, 30 years' time, you can still smell some of the smells that will disappear because of climate change in Rome. And the last project I will share with you today is a project we're doing with Maison Zero. Again, ecosystem thinking, really looking at this is working in the Amazon, working with LVMH and UNESCO, UNESCO Paris and UNESCO Bolivia. We're working in biosphere reserves in Bolivia, looking at helping communities to develop a more regenerative approach to their practice. Working with two communities, but I'll only show one today for conclusion. So this is a group of indigenous people called the Chiman. They live in the Pilon Lajas biosphere. It's the beginning of the Andes in Bolivia. You can see Bolivia there. It's just really at the edge. So it's quite mountainous, but it's just the beginning of the Andes. And the Chiman people will live, they have their houses. You can see you've got banana, you've got cotton growing, you've got cacao, but they are really surrounded by a lot of climate change related issues, a lot of wildfires, but there's also a lot of cultural issues. This is an indigenous territory. It shouldn't be used by anybody else, but there's constantly a lot of tension, people, burning forests for cattle ranching. So there's a lot of kind of local tension. But this particular village, this is Abraham, who has developed a whole agroforestry system to grow cacao with oranges and banana. So they're very skilled at growing plant food systems in agroforestry principles, which are very good in terms of biodiversity. They have a very artisanal production of cacao, and the women will still have, this is a traditional Chiman loom, so you can see this is the way they weave. So they still spin traditionally, they use local beads. But as you can see here, this is not the traditional Chiman fabric. This is synthetic dyed, again, fabric, yarns. And they're also really struggling to earn any living from this. There's hardly no tourists around there. And they've lost their knowledge. So we worked with UNESCO. We did a lot of surveys in the communities. They don't even know how to do natural dyes anymore. This is a Chiman indigenous group that are really traditionally close to nature. They live in forest, and they're losing their knowledge. So we were brought in to really look at how can we help and how can we develop the practice of regenerative design to help regenerate biodiversity, address the local climate issues, but also think about the social cultural challenges. I won't go in depth around this, but what's really critical for us, so this is a combination of surveys, scientific research, we have a lot of literature information, we know species that are endangered there, but we're looking at the local issues, the regional issues, and the global issues, and how they interconnect. And by analyzing all the different issues, so for instance, what we knew there from their literature, from even the Chiman literature, is there was a reference to a brown cotton. The cotton that grows brown comes from the Andes. They had lost it. They didn't think there was much of an interest for it. And we found it not too far and still in other villages. And we said, well, it'd be interesting to bring this back. This is part of your culture. We reintroduced the notion of natural dye using food waste. So if you use avocado skin, for instance, which they grow, you can get a pink. So even if they'd lost their knowledge of their natural dyes, we could reintroduce some knowledge, which was based on their food waste. In terms of climate change, moving more into agroforestry, they're constantly hit by wildfires, and so we want to help them to replant the fields, the forest that has been destroyed, with agroforestry system, combining food and textile agroforestry system. And in terms of the social cultural challenges, we're helping them to reach craft stores in La Paz and in Santa Cruz so that there's a real outlet for their textile production. But it's really looking at replenishing genetic biodiversity by bringing back that lost brown cotton, looking into developing more agroforestry system, renewing, regenerating their knowledge, their heritage, and developing a whole range of products. So what we did is we developed ideas based on their very special technique, their Chiman technique. But what we've done is developed ideas to propose to them to look at what would be of interest for them, but to develop products that would be more relevant to contemporary markets. We did a whole market research looking at what sells, what doesn't sell. So you have phone wraps all the way to bags, bottle holders, but also combining on the right, you can see two different weave techniques here, so which normally they do separately, and we've combined it into one product to give them an edge compared to other Chiman groups. We went there, we conducted a whole workshop, we really looked at what do you think, do you like it? They've asked for help, so we haven't been there to impose anything. This is a community saying we need help. We're not able to sell anything, it's not working. So we're redeveloping these very regenerative strategies that will benefit biodiversity because we're replanting more agroforestry. We bring back the cotton. We found the cotton. This is a neighbor village. They'd lost the plant had died, but they still had the seeds. So we're using these seeds to replant this in agroforestry system. And then we're really looking at, and this is an extraordinary, we don't have time to go into it, but their way of weaving, they use these little sticks to control the weave pattern. This is something which is very poorly researched. That knowledge is disappearing. So this is one of the things we're looking at now, is trying to preserve that heritage, because it's actually not documented very well, even just to know how to do this. So we've done a lot of work with them. They're now excited, they carried on. Since we've left, they've gone back to rediscover their natural dyes. They've relearned how to do the yellow. That is one of their local plants. But what you can see here is just a very different time scale. We started working on this with Maison Zero and UNESCO in 2021. And we're still at it. There's a slow pace working in terms of regenerative design. Whereas in biodesign, you can work fairly quickly over a matter of few months. With regenerative design, if you're really looking at having an impact on biodiversity, building that legacy takes time. And so this is where they are at now, really practicing, developing to actually bring back their traditional heritage. They've planted the brown cotton. It'll be ready for harvest next July. So we'll see where we go in July with them again. And I want to conclude with this final quote, because I think it's important for me that we don't think about our success criteria as designers in relation to, is our client happy? You know, if you're an architect, are we selling enough? Are we, financially viable? I think it's living system thinking requires that we think about our success in relation to how many living things benefit from what we're doing, humans and non-humans. And this is a quote from an ethnobiologist and farmer, and I feel that this should be really central to all the different design practices. I will conclude here now. I hope this was enough to inspire you to live, to share with you little seeds, insights and thoughts and inspiration for future practice. And again, we're after that systems change that we can adopt via a living system thinking approach. Thank you very much.
Thank you very much, Carole. Does it work? Okay. Thank you very much for your talk. I ask you to stay on the podium. We will now have time for questions, discussion.
Thanks for an inspiring talk. Many questions come up, but I would like to pick you up on the last project, working with the indigenous community. Are there any patents, copyrights involved in the project, and how are you handling this? And who would you consider to be the author, designer of the things you do?
So we don't own anything. It's all for them. Sorry.
But is this ownership, the collective ownership, protected in some way, or is this just an open source and open to appropriation by anyone?
So there are quite a few Chiman groups in Bolivia, and they all have this weaving technique is quite true to them. There's variations across different groups. It's poorly documented. but it is acknowledged as a Chiman technique. The products we're developing with them, so it's more co-developing. We wanted to kind of show them inspirations, other ways of thinking. It's very difficult. For me, it was a great learning curve because, you know, you go there, so you saw Abraham, who is in charge of the agroforestry there, studied agroforestry in Santa Cruz, so has left that village in the Amazon to go and study. So he's been exposed to quite a lot of a concept of IP in Chiman culture doesn't exist. Patent, they don't even know what it is. You know, you can't say, well, I come from France or from England. You come from the other side of the river. So you have to really adapt with the cultural language, the cultural way of talking about things as well, so that it's not overwhelming for them. But what we are now working with them to actually protect a little bit their ideas. So we're working with them the whole brand strategy, developing an Instagram account, to activate a lot of knowledge, to really make sure they are credited for this. And interestingly, the craft stores we're speaking to, they want a label. They want a recognition of the community this comes from. So we're developing this as well. So I think there's an interest in this, but we're not doing it in a Western-centric way. We're doing it in a way that's respectful of their culture.
Thank you. More questions? Helga, please.
Okay, so thanks a lot for this very, very inspiring talk. I was particularly intrigued by the microbial dyes. So I think there's a lot of potential there. And I have sort of a twofold question. So one is that sometimes natural dyes, they are not as stable as synthetic dyes. So is there an idea about the long-term stability, photo bleaching, and so on? And the second question is more into, second part is more into a design direction. So you have shown these very nice prints. Do you think there is potential in somehow co-cultivating these microbes with maybe other microbes, strange or fungi, to have sort of naturally evolving patterns?
So two very good questions. So the microbial dye for the PhD project with Colorifix, with Ruth Lloyd, this is based on Colorifix research. So what was exciting for Ruth is that she could work with us at Central St. Martin's, access to our print room, our grow lab, but she worked at Colorifix itself, where they're set up for all this testing. And this is the challenge in changing the system from synthetic to microbial dyes. And what they're doing is a systematic testing. So she's only worked with the blue and the pink because these are the two microbial dyes that have been systematically tested for light fastness, color fastness, to match like for like a synthetic dye performance. Because the industry will not adopt something that's a lower standard than the synthetic dyes. The problem with synthetic dyes is they're super cheap, they are versatile, you can create any color. So to replace that system, you need to get the same price point, you need to get the same performance in terms of the technical performance, color fastness, light fastness, and you need to start to grow all the colors as well. So it's going to take quite a few years before we reach that level, but this is why Colorifix is systematically testing to pass the test. Otherwise, there's no future for it. It's not going to work. And the second question was co-cultivation. Yeah, this is really interesting because thanks for asking that question. Often we don't talk about, so of course, for instance, if you work with the microbial dye, so in the case of the automating machine, so the fabric is actually sterilized before to go in, otherwise you get contamination. And so quite often when you work in biodesign working with one organism, you have to isolate it because you have to control contamination is often an issue. However, in nature, you have these ecosystems of bacteria. And I remember, and being French, I think I can talk about cheese, seeing a documentary that showed how if you're producing cheese in a stainless steel, sterile environment, actually you have far more chance to have any tiny thing that falls into it, you'll have suddenly quite a dangerous contamination. If you work in a traditional wooden kind of container, which is full of packed with a whole range of microorganisms, they will keep in check the more toxic ones. So I think that's the next step for biodesign, is to start working away from that sterile system into working with the ecosystem. It is quite difficult because particularly when you work, if you work in a lab environment, you need to be careful you're not growing something that's contagious, or that's, so there's a lot of health and safety around that. However, I worked on a project I'm not showing here with two designers in Paris called Alea, and they're really interested in looking at working with mycelium in soil. So they're designing chairs grown in soil. So they're actually creating a system to actually grow a structure within the soil using mycelium. And I collaborated with them on a project called Made in Soil, looking at how the textile design of, basically it's a textile cast, can influence the form and the shape and the patterning of the surface of that. So look up for them. They're called Alea work, and they're really looking at that ecosystem thinking, working with mycelium. I nearly put that in there, and I said I won't have time. So
Thank you. Clemens had another question. Clemens Abrich.
Hello,
Oh, sorry. Okay.
thank you very much for a beautiful lecture. My name is Natalia. I'm a local fashion designer, and I'm wondering, do you see any visible perspective for replacing outerwear materials with something natural? As like for now, we have only mostly polyester base, even if it's like whatever bio or not, and also like something where we can move away from a Gore-Tex and similar technologies, what brings only harm for environment and also for humans who produce it. As like we can replace down, for instance, with this plant fur, but how we can replace the actual fabric, what keep this down? Thank you.
Very good question. So if you look at 120 years ago, none of these fabrics existed. The entire textile industry for millennia was natural based, the wool, the linen, the cotton. And then the 20th century just shifted completely towards a petroleum-based industry. So we've now developed these high-performance polyester, polyamides that are very water-resistant, or we developed these, a cotton coated in these forever chemicals, which are extremely toxic for our health. And we've become used to it. It's the new normal. So now to shift back to doing it differently is kind of a bit of a mindset as well. So there's two ways. Either you look back, actually wool can keep you warm even if it's wet, but it's wet. But I've seen recently a few interesting innovations. So Laura Piana, based in Italy, part of the LVMH group, have developed a bio-based coating system called BioStorm, exactly for that, the outerwear performance. I read actually even just last week, I think it's either in Canada or in the States, a research group developing a mycelium-based coating that's hydrophobic. So this is coming. See, right now what we've done with biodesign and biodesign research in the textile fashion industry has been mostly material and color. The finishings is really the key, and I'm starting to see companies engaging with that side. So the waterproofing, the water coating, and that's going to really start to change the system. But I think it's going to be another 10 years before you really get them as the new normal, and it's going to be a cost issue again. Yeah, but I have hope.
Thank you very much.
Thank you. Clemens, please.
Thanks. Thanks also for as you said, the diversity of approaches. I'm not a designer. I'm also not an architect. I come from media theory. So I want to go back to one of the premises you said, when we move from this anthrocentric, paradigm to something as a new paradigm. Do you see like a tension or like a danger also in, anthropomorphizing this kind of like natural, approaches that you say, nature itself? You said like, nature as a manufacturer, but also like we should not refer to the maple tree as it, but she. There already comes like a gendered relationship into it, like Gaia as Mother Earth, as a caretaker. Doesn't this confine you? I mean, this is like a provocative question, but does it confine you as a designer also in the approach to nature when you also showed these contingencies in nature, something which is maybe alien, right? And not like a kind of like a human attribute that we put to it.
I think this is a very good point. We can't romanticize nature. I mean, nature is pretty violent. and was I mean, you can go for a walk in the bush in Australia and not come back, so I think we need to be careful to not think nature is beautiful, wonderful. What we need to look at is what we can learn in terms of systems and behavior that can be really interesting for us. And we discover new ways of doing things constantly. I was reading actually this morning before to come over, a research group in Harvard. They've studied a slug you find in the ocean, which looks like a lettuce leaf. And it's one of the only animal we know that actually can do photosynthesis. So it's crossed that divide. How does that work? And so I think for me, it's really looking at learning from how does it work. It doesn't mean that we should put nature on a pedestal as this is, but it's what can we learn that brings that circular thinking so that whatever we make doesn't have an environmental impact, but actually the waste of what we make is a nutrient for another set of species. And that's the principle of, the circular economy is based on this sort of natural cyclic system. But I think for me, it's important to also really look at that notion of kinship. Because if you don't really value if you don't value your person next to you or your friend, you're not going to really respect them necessarily. You might use them. So I think it's really thinking about how we can act in a way that is respectful towards other species. Currently, we are exploiting species. I mean, if you look at particularly the agricultural system, the food system is extremely exploitative of other species. Exploitative of farmers too, I would say. But I think we need to be careful to not fall in a romantic kind of, beautiful nature, but really looking at what we can do to get a step closer to how nature works.
Thank you very much. We're now going into the coffee break, and you will have the opportunity to ask more questions, and we will continue the discussion with the first session in 15 minutes at 11:15, Design with Organisms. And I would like to ask the session speakers and the session chairs to take place in the front row when you're coming back. And please enjoy some drinks. There's also the Mensa outside for you to frequent. And see you in 15 minutes. Thank you very much.
So the first session is called Design with Organisms. And just to introduce myself shortly, my name is Orbanić. I'm a designer and researcher, and I myself also work with mycelium. And therefore, I'm very excited to be here today and welcome all of the guests, as I know it's a very exciting and very thrilling, very challenging work as well. And I'm very excited to hear about all the knowledge that you're bringing with us today here to Vienna. And thank you for being here. And as I might have already given a small hint, it's not only about design with organisms, it is also about a specific organism, which is mycelium. And I have a feeling that most of you probably in the audience know what mycelium is, but usually I like to give just a small introduction, since I like everyone to have an overview of what we're talking about. So in case someone is not aware, mycelium is the vegetative part of a fungus. And one can imagine that as a root-like structure of a mushroom, which commonly grows underneath the surface, so in the dark. And it's a very fascinating organism. It has a lot of interesting properties, which I'm sure we're about to hear about today. And but one thing which is truly fascinating is that it digests organic matter, and it's very crucial for our environment. And by doing so, by digesting organic matter, it itself as an organism grows bigger in size. And coming now to the topic of design with organisms, or design with mycelium, it is really interesting to think about it, because not only it's a very important topic for the circular strategies, the future circular strategies and ways of future designs, but it also challenges us as designers to rethink what design is and how one can work with organisms. And this very much means we have to collaborate. As in every type of work, here it comes to the point that we have to collaborate with the organism itself. We have to understand which conditions it needs, and sometimes it even means we have to give over the control to the organism itself. And what we're about to hear today are 4 papers for research and in-practice projects developed at 4 universities, which all work, explore, and collaborate together with mycelium and experiment on different mycelium potentials. And I would like to give you quickly just a short overview of the presenters and the presentations we're about to hear. It will be 4 presentations, so there is 1 person missing, which is Anne-Kathrin. It's not on the slide, I'm sorry for that. But to go through the presenters, we have Natalia Piorecka from the University of Innsbruck, and with the paper titled Extreme Matter, Mycelium Composites Across Material Regimes. Afterwards, we will hear about a presentation from the University of Stuttgart, presented by Eliza Biala, with the title Solidified Elasticity of Mycelium-Textile Hybrid Architecture. The third presentation will be presented by Evgueni Filipov from the University of Michigan, USA, and Newcastle University, UK, with the paper titled Bonding Mycelium Biomaterials with the Built Environment. And lastly, from the presentation from Anne-Kathrin from the University of Stuttgart will be Entangled Mycelium, Textile Logic for Grown Architecture. And finally, I can invite the first presenter, Natalia, to please join me on stage. And
hello, everyone. Thank you for the introduction of the mycelium. It's going to make my job a little bit easier now. It's a pleasure to start the presentations today. And I hope everyone's going to have a wonderful symposium. So my name is Natalia Piorecka. I'm representing a team from Integrative Design Extremes Research Group at University of Innsbruck. And today I'll present our kind of ongoing research called Extreme Matter, Mycelium Composites Across Material Regimes. And it's part of a bigger research project called MUSE, Mycelium Seed, where we really look at advancing mycelium-based materials, broadly speaking. So to kind of start with, I think it's important to picture the extreme context that we're currently in. So the material challenge is real, the material crisis is also real. Unfortunately, some of these things are not as obvious as they seem. So in case of this crisis that we're focusing here, the sand. So over each year, more than 50 billion tons of sand, gravel, and aggregates are actually excavated. And out of that, 70 to 90% are going directly into construction industry. It's important to mention that not all sand is the same. So not sand could be usable. So the desert sand is too smooth, the beach sand is too salty, and of course, extraction harms the environment. So what happens is that the construction grade sand is actually mined beyond the recovery currently from rivers and quarries, depleting also the natural landscapes. In 2023, United Nations Environment Programme declared sand crisis, really kind of putting that matter publicly. And of course, what happens with the sands, with the construction grade sand, once it's used in the concrete, it becomes inseparable. non-recyclable, and most of it ends in landfills together with the rest of the construction waste. So bringing the protagonist, mycelium, in board, the question is maybe how. So we know what mycelium is, the roots of mushrooms, the principal decomposer of our planet. This is also the processes that we use to create the composites with the organic matter, so the degradation, the decomposition. But the question that we want to ask here is if this living adaptive binder could also stabilize inorganic matter. So we've been referring to this approach or strategy as encapsulation, not in a chemical sense, but more of a physical sense, where the physical enclosing of one material happens with another. So really trying to kind of see if mycelium could grow around. I mean, you can see some of the examples in the diagram and the microscopic picture of how potentially mycelium could tangle around the inorganic matter and stabilize it. So with this in mind, we've been kind of working with two parallel techniques. So the first one is the kind of probably familiar to you as well, the decay-driven strategy with organic, which is purely based on the biological degradation. So in that case, actually, the substrate is the nutritious source for the mycelium, and through the processes of decay, we kind of achieve quite lightweight composites. And then the other strategy, the encapsulation, really works with the physical encapsulations, where the nutrient is a bit problematic. So one always needs to remember that it's a living organism, and it requires a certain amount of nutrients. Therefore, there's always a little bit of a mixture in there. But what that allows to happen is that the mycelium kind of seeks for the nutrients around the inorganic material. And what's interesting about it, that the potential of it is that potentially we can have a longer durability time for these materials, what opens also new applications that were maybe not really tangible before with the decay-driven material, which is destined to decay. So kind of looking a little bit up close to the kind of binding logic. So really at some point, because the mycelium grows inter with the substrate, really decomposing it partially to a certain extent that at some point with the growth, you can't really distinguish the mycelium from the nutrients. It really becomes one mass, where again the substrate is the nutrition source. Whereas in the other part, again, this is the mycelium that needs to look specifically for the nutrients. So one can really play it with almost like a lattice structures that it's locking different kind of materials. And just as a comparison, so the first one really kind of goes, we can use agricultural waste, we can grow, we can use, it's destined for a decay as its kind of end of life. Whereas with the other one, we could actually tackle different kind of waste. We can grow, we can use, and potentially actually also regrow and reuse, putting the materials that are not currently in the loops, well, in the loop. So some kind of inviting the circular aspect here. And in terms of the material development, one needs to balance the inorganic aspect and the nutrition sources. So the kind of composition and the material methods of growing are kind of key here. And for example, yeah, it's a little bit moved, but pretty much what happens when you have small particles and very low amount of nutrients. We've been working with something that's called, that we've been calling sequential layering colonization, which really allowed to kind of fragment the fabrication process, so the mycelium can get enough oxygen on each of the levels. So it was just too densely packed with these two small particles. So in general, dealing with the sizes of the elements, the kind of how we produce this material was something that we were trying to tackle over here. And some of the preliminary results, I would say, is that the sand-based samples were over twice the compressive strength of all the tested organic substrates. So we're looking from sawdust, different mixes of fibers, hemps. That also got reflected in terms of the weight, of course. And then the substrate became from 3 to 5 times heavier than the organic ones. So we suddenly changed completely the kind of principal material property of being extremely lightweight. In terms of the fire resistance, both organic and inorganic, they resisted the ignition. Then what was interesting actually about the sand is that even in the prolonged exposure to fire, it really didn't show a lot of change on the surface of the material. And with keeping in mind this kind of shift in material characteristics, we can also ask what kind of different opportunities, different kind of materials could be embedded using this principles of encapsulation. So we can also look at different other sand-like matter. So reaching the desert sands or construction waste, crash bricks, different mineral waste, even also extraterrestrial matter such as regoliths. Then another question is we can actually use different offcuts from different composites that maybe are difficult to recycle. Maybe they're already mixed with something. Another aspect, I'm sure you're going to hear a lot about textiles soon as well, but the synthetic textiles, can they also be part of that? Can they be kind of embedded temporarily in the material? And also kind of foams, insulation offcuts. We've seen that a lot on the construction sites, that it's a big trouble. They're not really recyclable in that sense. The big question is what kind of materials could be actually safely encapsulated in this case? What are the implications? And how could we integrate this encapsulation, this temporary containment, into the circular construction systems? So it's not a finished project. It's more, I think we've been thinking of how to evoke some thoughts, and I think we're very excited for discussions later. But the questions would be like, what actually happens with the encapsulated matter? Do they remain stable? How long does they remain stable in different environments? Can these composites be disassembled and assembled? Can they be reactivated, regrown, or reconfigured? And really, probably a big question is, are we delaying a problem, or are we giving it creating a new value for the waste? And then broadly thinking, how can maybe different biological processes be integrated as part of the system? So maybe even more extending the longevity of these compositions. And something that's applicable for both of these materials, for both organic growing and inorganic growing, is the consideration of energy demand, of the inoculation, incubation, but also the waste treatment. So can we actually look at local wastes? Maybe this is just a technique and we kind of always work with something different. So how can we adapt it to the context that we're in? The regionality of the fungal species, of course, is very important. How do we not disrupt the local biodiversity? The mycological research, as actually some of the specific fungi might decompose these inert materials and break the hazardous parts into something that's less harmful to the environment. And how we're dealing with the second life of these inseparable waste streams. Is that recycling, upcycling? How does it feed into the bigger picture of our economy? And what are the implications in terms of material characteristics, application? Can we, for example, start talking here about indoor and outdoor, and outward applications? They're using sand in different, completely different contexts, completely foreign context, as an in situ resources. And I think I'm actually right on time, so I want to thank you for that. And I think I'm hoping that some of these questions will evoke some interesting discussions. The team is sitting around everywhere. We also have our samples on the other side of the room, so please have a look. You can also touch them and smell them, and we can actually probably have very nice discussion over the samples. So thank you.
Thank you very much, Natalia. We can take 1 to 2 quick questions from the audience before we jump to the next presentation. Yes.
Hello, thank you very much for your super interesting presentation. I would like to ask, in which percentage the inorganic matter is within the mixture?
Sorry, can you ask it again?
If it's like 100% inorganic matter in this mixture, it's like
for the organic samples. Yeah, I mean, we work with different mixes, but usually it's organic. It's usually one fiber. I think we really kind of wanted to start building a material library so we can reference these things against themselves. So it's it's usually one type of fiber.
Good. Thank you.
Yeah.
Hi, thank you for the presentation. It was really, really interesting. And actually my question is very similar, but about the inorganic composite. Mm-hmm. Is it 100% inorganic? Is it a mix? How much?
Yeah, so in this case, when I mentioned the kind of talking about the composition, you need to have a source of nutrients for mycelium to grow. Yeah. So the question is, okay, what kind of nutrients you provide and how? So through this research, for this particular study, we're looking at different proportions. So it is always a bit of a mix at the moment, just to kind of allow mycelium to grow through. And it's also somehow, you know, when you have a strong colony of mycelium and you put it in something that maybe is not clean or it's not sterilized, so you avoid this step of sterilization, then if the mycelium is strong, you can still take it over. So that was kind of the starting point for that. So when you put some sands, which let's say are not sterilized, you kind of slowly grade how far you can push it. So I think this research was really trying to see how far you can push it and what are the implications. And also this is also the sequential layering, because it was pushed a little bit and it needed a bit of a different fabrication method for that.
Yeah.
Does that answer the question?
Yeah, no, absolutely. Also amazing to know that you didn't sterilize it. That's great. Like,
it's I mean, it's a lot of energy. So
yeah. Okay, thank you for the answer.
Thank you.
Yeah.
Next, I would like to introduce Eliza Biala, who together with Martin Ostermann developed a paper at FUMA Lab at the University of Stuttgart, Germany, entitled Solidified Elasticity of Mycelium-Textile Hybrid Architecture. Welcome.
Oh. Okay, so thanks for the introduction. I don't have to present our first slide. So the research I would like to present today aim at developing scalable biofabrication process for creating doubly curved mycelium-based composite panels with the use of elastic textiles as the lost formwork. So something wrong? Okay, now it should work. Okay, so mycelium-based composites are the genre of materials which actually can be designed. We can do it with twofold goals, either to adjust the material performance, either to develop new fabrication methodologies. Textile hybridization is actually possible with mycelium composite and can profit from both of those goals, either to contribute to targeting certain material properties and predominantly improving the tensile strength of the material samples. Either it can unfold the creative approaches to new ways of building and fabricating with mycelium composites. So textile hybridization was a big part of the works in FUMA, which were done since it starting up in 2021. So we tested it in many different approaches, and we have this focus and we want to continue it in the following semesters and months. So the project I'm presenting here was actually driven not purely by the curiosity, but was developed within the framework of the Design Commission, which came to us in the December last year, when through our partners and collaborators, it turned out we could exhibit mycelium flying installation in the gallery in Venice, which will then stay there for half a year in parallel to the Architecture Biennale. So between December and January last and this year, we were working with the curatorial team to develop the border condition for the installation, which then could be realized within the student workshop, which were planned for February this year. So in this way, we were going through different samples and different approaches we had so far in FUMA, and we were wondering which ones are the most promising and fresh and actually possible to implement within the given framework, which, as you can notice, was very tight, and as well as how our budget was also tight due to all these time conditions. So one of the samples which came to our focus was very small one, which was actually applying mycelium paste on the tensioned material. It was very small and was developed in a framework of master thesis project of Kalayamuthan, Alizo Ghaffari and Magdi Ghaffari. But it was one of the paths which they decided not to continue and to abandon. But for us, it was a very small proof of concept that actually elastic deformation and tensile structure could be solidified to self-holding stiff surfaces with the use of mycelium paste. So we decided that we want to take this approach, but not to think about the textile as the pure form giving or the pure forms. We just want to like stabilize, but we really wanted to investigate the interconnection of the growth and of the mycelium cultivation process, to work with the forms which actually support each other with the materiality, not to work against it. So to make whole project realizable, we had to design some border condition of what we are working with. So first was that within those first 2 months of like looking on it, we discovered that the textiles really not to have like creases and problems with fall development have to be knitted, not woven. Plus our budget restraints put us in a situation we have to work with second-hand textiles, so actually be a form of the textile reuse. We initially wanted to work with the first-hand material, which we eventually got, but actually after the workshop was finished. But we got saved by our research partners from other project from German Institute of Textile Research, who is actually positioned very nearby from our laboratory, who gave us actually some leftovers from their machine tests, which were in their basement, and which were then the materials we were working with. The second border condition was the initial material. So we pre-grown the sawdust with Ganoderma lucidum to have like a starting material with which we'll be working later on. To change its consistency for very particular to paste-like, we used the additives of guar, gum, and wheat in different proportion, which was the variation of the recipes which we were employing earlier while working with mycelium 3D printing and filament extrusion, which Kathrin will present in 20 minutes. So the next border condition was the cultivation setup. We knew we'll be using big IKEA boxes, which we had in lab and which we used earlier for growing mycelium samples. And all the scaffolding tension structures were supposed to be built from the things which we already had in the lab from earlier design and build project developed by the university students. So at the time we thought it's going to be mainly styrofoam, which we have plentiful due to some earlier activities of the institute, but it turned out it's actually not suitable, but on some samples you still can have. So the first part, the first week of the workshop, was design research into the modularity, which can be easily cultivated and which have certain architectural values. So it's like creating surfaces, making three-dimensionality, working well with how the material covers it, working with different cultivation strategies. Basically, we try to develop on one side the geometry, which could be aggregated and which could be growable, and on the other to develop cultivation protocols which will be easy to multiply. So for all the initial tests, we're creating protocols where we exploit different aspects and try to record different aspects of the process. So initially we tried with the sterilization only by the mean of the water bath, and then depending on what elements of the scaffold were there, we were either going back to the very standard sterilization with the ethyl alcohol, either we were omitting it. Also different thicknesses of the paste, different compositions of the paste, and different materials were tested and recorded. So each test was actually informing the subsequent one. So it's not like we tested everything with one configuration. As you see, there was really 20 parameters which we can variate. And actually the moment, the feedback loop was kind of long. We knew we work within the framework of 2-week workshop, and growing takes time, drying takes time, and some things can only be noticed at the end of this period. So I have few snapshots of different aspects which we were discovering on the way, and then I will sum up all of them together. So the first formulated issues were we wanted as much of three-dimensionality as we can, and for architectural reasons we are making first a lot of tests with the continuous edges which could be aligned to each other. The second type was working with the point attachments, as they were much easier to fabricate and were also easier to demold. So what edges showed us very fast was that the stiffness of the edge is crucial, as we tried to build edges either out of wood, either out of willow, either out of miscanthus, either of glass fiber. And it turned out that the wood is too stiff and biomaterials are not creating the twigs are not creating the very smooth lines which could actually fit with architectural arrangement. Plus, creating the attachment was super problematic. In most cases, it leads to cracking, though like the last example with willow and with a bit different composition of the paste was actually working, though it was very requiring a lot of time and effort to even make it possible. With the edges, on the other hand, the problem was cracking, so that the material couldn't go till the end, especially as it's very thin with our approach as applying paste. So creating a bit of spacing before the edge was really helping with the demolding and to taking away the material away from the form-giving scaffold, though it was creating the point where the contamination could appear. So molding points were the observation, which was more like coming from the observations of different tests. So the wooden sticks turned out to be not suitable, as the place where they were getting the more wet was always the contamination point, the same as the wooden blocks. And the main character of this fabrication method was that after 2-3 days, we have a lot of water release, which is not really present when we work with mycelium composites without paste additives. So to sum up, we discovered that we need textiles which have like really big stretchability, able to withstand the high load of the paste. The scaffolds has to be not porous or sealed to withstand high tensile high forces, and to get the homogeneous expression of both sides, we really need to add a bigger content of flour to boost the outer growth and therefore the aesthetics of the panels. So after the form was decided, we moved to the production phase, where we started to repeat the so-called bird form, as we named it. And we did it with around 20 students in FUMA, which was nicely recorded by the vlogger flatmate of one of the students, so you can still see it on YouTube, and to get a bit of the vibe which we had there while producing all the modules. So all the rooms we had were started with boxes, which then had to be always wiped up. They were growing for 4 days, then they were moving to drying. During drying, we discovered that actually not covering the edges helped not only with the molding, but also with shrinking. You can see the angle change of the textile. And another, that what we didn't expect, that actually making textiles dirty with mycelium are the best way of protecting it from contamination. Like wiping with alcohol doesn't help, but having the growth and actually our organism taking place first, it's much more efficient and results in a very thick layer of mycelium foam growing directly on the textile, almost without the substrate. We had also a few things which we noticed just on the few panels, not on all of them. One was creation of water blisters, which were actually this released water from the paste, which was no way to get out, as the skin was already so thick and it was protecting it, though they were drying out and there was no issues with it. The second surprising occurrence was actually the second growth of mycelium on top of the skin, which we still actually don't know if it's the unplanned co-cultivation of two organisms, or it's just the different type of growth happening on the surface. We tried with repairs, they didn't work as contaminations comes in the intermediate zones, and we managed to grow almost 600 panels, which then were applied in the exhibition, and the uniformity was allowing for making pretty consistent and covered surfaces. So our main bottleneck was actually drying of all the panels in the Räumlichkeiten, so like the space we had available. So installation then survived half a year in Venice, Plus, it survived then the way back to us, to Stuttgart, where we dried it very intensively in our rooms once again to actually take away the moths, which actually started to live in few of the panels, from not all of them, like maybe 5%. We hope the heating it up for many days in 50 degrees really allowed us to get rid of them and have the installation ready for the new adventures and new installation points. So the whole research was done during the first 2 weeks, and it happened together with the students of 2 universities, Hochschule für Technik in Stuttgart and University of Stuttgart, and they are like here all on the photos. So one of the modules will be exhibited here as well. It will come during the lunch break. If you would like to touch it or have any more questions about either the material, either the process, I'll be happy to connect and to exchange in this subject.
Thank you, Eliza, Biala. Please, I would ask you to keep your questions for the end, since we're a bit more tight in time as expected. But as the next, so we will come back to the questions at the end. Thank you. As the next presenter, I would like to invite Evgueni Filipov. Thank you for being here, from the University of Michigan, USA, and Newcastle University, UK. He developed the paper together with Ben Bridgens and Jane Scott from Newcastle University, UK. The paper is titled Bonding Mycelium Biomaterials with the Built Environment. And I would ask you to take over. I think you just have to click over there.
Well, first, thank you so much for the kind introduction. And I'll start by saying I'm an engineer, so maybe this is not my typical crowd, but I think it's actually a very good place to present some of the recent work we've been doing. So I'm Evgueni Filipov. I'm at the University of Michigan. This is work done with some of my students, by Babi Chidala, Kwang Wu Yi, and then at Newcastle University, Ben Bridgens, Jane Scott, and Rami Kaiser. So a little bit of a history about me. So before 2023, my work was quite different. At the University of Michigan, I run the Deployable and Reconfigurable Structures Lab. We focus on origami-inspired structures. I've worked a lot on developing simulation tools and models for origami about how they fold, how they deploy, how you can apply loads onto them. I'm an engineer again, so looking at can we build these and can we make large-scale applications for use in infrastructure systems that are lightweight and can carry large loads. And then at the same time with collaborations, I've looked at micro-scale origami-inspired structures where you can look at miniature-scale systems that can interact with matter. And in 2023, I got tenure, and that got me thinking a little bit. And my work is great. I still do a lot of origami work. I think it's very fascinating, but there's been challenges that my work doesn't address well. So we all know 37% of global greenhouse emissions come from buildings and infrastructure. And in particular, embodied carbon, right, the things that are around us, are particularly hard to decarbonize. While we can decarbonize and we can electrify a lot of things associated with heating and cooling infrastructure, that embodied carbon is particularly difficult. That's when I met my hosts at Newcastle University. That's where I'm currently doing and finishing up my sabbatical. So in 2025, I worked with Ben Bridgens and Jane Scott, and their exciting work on mycelium biomaterials that we've already heard a little bit about, right? But can we use growth to replace construction? Can we build with agricultural waste versus mined materials? Can we sequester carbon dioxide in infrastructure, maybe create self-healing materials and so on? I think the applications are broad, right? We can have packaging, furniture and products, architectural systems. And I should acknowledge this work is funded by the Simons Foundation. This has allowed me to go and work in Newcastle. So when I went there, I decided, okay, I need to have a structured goal and I need to look at something. So I decided, how does mycelium bond with other materials? And I think this is actually a nice segue to some of the previous two talks we've heard already. I decided, okay, I'm an engineer. I need to keep my problem as simple as I can. So we'll start with a very simple preparation process. We'll start with 2 kilograms of beech sawdust, 2 liters of water, sterilize that, inoculate it with mycelium, let that pre-grow for 6 days, break it up a few times along the way so that myceliums colonize the entire medium. And then we use these little coupons. So these are basically, if you think of what your ice cream comes on, basically the same size, 8 centimeters long, 1 centimeter wide, about 1 millimeter thick. We put these coupons in these little trays. let it grow for 15 days. You come up with this beautiful thing here where now the whole thing's colonized by mushrooms. And then after that, we decide, okay, can we test this? And what we do is we put this in a vertical testing machine, an axial testing machine. We pull it up with the force. At some point, you get this to break. And now I'll show you over here. This is most of the technical things that we'll be showing here. But the pull-out stress. So basically, we're normalizing that force by the surface area. So if we have coupons of different sizes, we can compare them in a logical way. And so you see in the very beginning, right, over here, 0, up as we break the bond, and then after that, it goes down to 0 at about 440 millimeters, right? So once the coupon's entirely pulled out. And I have the results shown here for different days of growth. So day 0, basically, if you pull it out without allowing the mycelium to grow, that's a flat line here. The sawdust doesn't really provide any strength. And then you're going up and up and up to about 25, maybe 30 kilopascals. To put it in perspective, that's about 20, 30 newtons that we're applying a force up. And that's about 2 to 3 kilograms of force. So that's not nothing, right? You've got this little tiny stick sticking in the mycelium. You can apply 2 or 3 kilograms of force that you're required to pull it up. So we looked, all right, what about different woods? And so we tested 7 different woods, 3 engineered wood composites, paper. And so here's the results. So for example, I'll show you this. There's some things. Paper, for example, breaks. That's why it's an open circle here. The squares, that's our 4 days. Circles, 8 days of growth. The triangles are 15 days of growth. But overall, everything, all these organic materials bond quite well with wood. Something that's quite interesting is this trend of mass loss, which is the opposite trend of what you see with strength. So these are arranged in the same fashion, but pine, oak, for example, which bond quite well, have a much lower mass loss than some of the things that don't bond quite well. Beech and birch over here. Okay. We think the reason for this is, well, oak and all these other materials that are a little bit harder for the mycelium to degrade, they still have nutrients. The mycelium's trying to get in there, so it's actually investing more resources in colonizing the area more densely so that it can try to get into those resources. Okay, we looked at other materials. Ceramics over here, textiles, plastics. And by and large, mycelium bonds with just about everything. There are some outliers, for example, latex here, very low bond strength, but that I would say is latex is very stretchy, so as you're stretching it, the debonding mechanisms are a little bit different, so that's why you get a little bit of much weaker bond there. Some of the other more interesting things to take a look at are the textiles. So here, these flat lines that I'm showing for these material groups, that's the average of the entire material group. So plastics, 20 kilopascals, not bad, not too different from the wood, 25 maybe. Ceramics a little bit lower, maybe 18 or so, 17. Textiles quite a bit higher, about double, okay? And so we're getting a little bit more strength when you're working with textile. And another one of the outliers in the ceramics here is this clay brick. And I just want to show you here, we've grown the samples against an acrylic, so you can see just the way the growth happens. And if you look here, this is our textiles. There's a little bit of a white boundary on the outside here, and on the brick as well. You see a little bit of a white boundary. And what we believe is happening is there's just more oxygen access, more nutrient exchange that's able to happen through these porous materials. In this microscope image, you see a little bit of denser growth happening right there, right at the interface between the textile. This is with wood, and you don't see that, right? So it's not a porous material, and you're not seeing that growth. Okay, so we know mycelium can bond well with everything. Well, this is pretty exciting. I think the previous 2 talks have shown that. Maybe we can use all of these materials and integrate them into our future biocomposites built with mycelium. And so here's a couple examples. So first, a beam, right? You can make a beam, you can reinforce it, thinking very much of reinforced concrete, some strips of material on the top, or maybe fibers on the bottom. These are the systems that have strips on the top and bottom, so much higher strength. The blue here is just mycelium, so doubling the strength just by putting in some reinforcement. If you have just fiber reinforcement, maybe not much strength, but it's this after behavior happening over here, where now, even after you've broken the structure, there's still some residual capacity. So that's exciting. Okay, you can make encapsulated systems. You can maybe make a system where you've got polymer on one side, so we use PETG plastic, so that's food-safe plastic. Basically, a lot of your food containers come that way. And then wood. This system here is wood and wood. This one's plastic and wood. And you can see here triple the strength of what you'd get versus a pure mycelium composite, just because you've now put those upper and outer layer, where now you're basically making a composite system capable of integrating with those other materials, not just the mycelium that you're growing and the substrate that you're starting with. Okay, well, if we can connect this with plastic. Maybe it doesn't have to be structural. Maybe it could be other type of materials. Here's what I like to call our window. I think this is a lot of fun that I've had with this little experiment. And so here we have again PETG plastic encapsulated by mycelium on the top and bottom and all around. Here it is as it's growing, demolded, a little bit of breaks. It continues, it regrows those breaks. You have to clean the mycelium off the window sometimes because it tends to colonize, looking for nutrition everywhere. Here it is, it looks out. You can see the buildings through it, some water vapor, and then we dry it. And I said, okay, well, we can test it. We can also try to test this. And so we decided our window can act a little bit like a cup. What if we pour some water into it? There's about 2 centimeters of water that end up getting poured, 180 millimeters. And I said, okay, well, I think it's going to leak in the first day or two. So we were measuring very frequently early on, but it's been 30 days. This experiment's still going on. It's still not leaking on the bottom. I would say we're not perfect yet. So yes, mycelium is hydrophobic. I think there's a lot of potential advancement that will happen into this to the point where we can get these to be fully impermeable. Right now, there is some absorbed water, right? So we measure the mass, and so there is absorbed water. We dry it fully initially. There's absorbed water happening afterwards, but that stabilizes. And then, the top area here where the water is, that's wet. The bottom area stays dry throughout. At day 7, we see a little bit of moisture happening up at the top here. And then on day 16, we do see water coming out at the edge. So I think at day 16 or so, maybe our experiment has failed. Okay, I talked about these 4 materials. Great, we can incorporate all these other materials, but there's one material class that I've left for last because this is the most exciting and I think potentially the future of where my work will go. Metals. And so we looked at metals as well. We looked at 10 metals, and I've grouped them into 5 metals that bond a little bit better, 5 that don't bond quite as well. The scale here is quite different, and just to put it in perspective, I put all of them together. So here's a ceramics, plastics, woods, textiles, about 2 kilograms of force that we require to pull up. The metals, on the other hand, are an order of magnitude stronger, okay? So that little strip of metal, you can have to apply about 20 kilograms of force before it pulls out. Okay, this was quite a surprise to us. This is not porous. There's no oxygen getting in through there. But there's quite a bit of a different mechanical behavior that happens there. And we're now trying to dig into exactly why. Here it are just tiny little particles, 1 millimeter or so in all dimensions, growing inside a petri dish with the sawdust. We have sawdust all in this medium. So we have mycelium integrated into this whole mycelium. It's not just in the middle. It's growing everywhere. But in those locations where we have some of these metals, we see very rapid growth happening right around the middle, around day 7 or so. So the mycelium finds this, and what we believe is happening is the sawdust, right? We've constrained it. We kept our problem simple, but maybe that sawdust is just not enough for the mycelium. It finds that copper, it's the nutrition that it needs, and now it starts going and colonizing that area, and that's where we get intense growth, much more higher bond strength that's happening right there. Again, very preliminary work at this stage. Here's our copper particle. Here's an SEM image, basically, where we're looking at this under a microscope. You can see, right, copper particle over here. If you go far away, so about 1 centimeter away, very classical mycelium networks over there. As you get close, much denser, and then very near the particle, intense growth. And what we think is happening is it's just trying to access that copper, which it needs for its development and its growth. But we think this is very exciting, right? Maybe what we do need to think better is how are we feeding the nutrition, our mycelium, when are we feeding our mycelium, and how are we integrating other materials as we do that. So I'll give you a quick summary of my work so far. Again, a lot of this is preliminary. It's just work that's happened over the last year. But we think mycelium is an excellent bond agent with a lot of different materials. We've tested 45 different basic materials and composites, and it's bonded well with all of them. We have good bonds with metals, especially the copper, zinc, nickel, iron, and their alloys. We can harness these for structures, and then we can hopefully integrate this into the built environment. And with that, I'll say thank you.
Thank you very much. We will come back to the questions at the end. Thank you for your presentation. I'm sure you all have a lot of questions, so don't forget them. And lastly, I would like to invite Anne-Kathrin. Thank you for being here, together with Eliza, Biala, and Martin Ostermann, which we have heard in the second presentation. She developed a paper called Entangled Mycelium-Textile Logic for Grown Architecture. It is as well part of FUMA Lab and is the University of Stuttgart in Germany. Thank you for being here.
Hello, my name is Anne-Kathrin. I want to present you now the project Entangled Mycelium-Textile Logic for Grown Architecture. I'm an independent design researcher. My background is in textiles and materials, and I've been working on the topics of solidified textiles for quite some years now, and the project I'm presenting is also partially built up on my master's project where I developed textile structures with concrete. I'm doing the research together with Eliza Biala. You just heard her presentation. And all of the research happened in FUMA lab of University of Stuttgart. The objective of our research is the architectural potential of combining textile construction techniques with mycelium-based composites, MBC, in the form of filament hybrids producing upscaled solidified textiles. So we envision structural applications of textiles in lightweight and biodegradable architecture. The research follows a practice-based approach, and a lot of prototypes were made. And I want to give you now a glimpse of the activities happened so far and dive deeper into textiles and show you the results of the Collet learnings. So textile techniques have always been part of architectural practice, from woven walls to tensile membranes. Textile systems do not merely produce surfaces, they define spatial logic, and textiles are actually made from yarns or filaments that are processed by a textile construction into surfaces. And the textile properties are influenced by a lot of various parameters, like the material, like the technique, or other parameters like the dimension of the yarn. And all of that has an impact on the, it all generates a different outcome of the textile. So when working with textiles or in textile production, you have to define various parameters. First, starting with the yarn property, with the filament, then the construction technique, and then the shaping and cultivation process, which ultimately leads to the final form and behavior of the textile. So starting with the filament production, for that we needed to make a filling paste first, which consists of sawdust and in combination with Ganoderma lucidum and water. And to achieve viscosity and to make the paste smooth and viscose, we added plasticizers. This is me first, when I started with this, was stuffing the sleeves by hand, and also university did. And then, because this was a crazy amount of work, the university luckily bought an electric sausage machine, which was a 100% improvement, because it was much easier to fill the sleeves with enough pressure. And as the skin, let's say, we used off-the-shelf textile sleeves. So here you can see the process of the filling the textile tubes with the paste, and the result is a wonderful evenly stuffed sausages with the mycelium paste. An important point to mention is that the sleeves are not removed afterwards. It therefore function as a lost framework. So next step is then the textile construction of the hybrid yarn. Only when embedded in these systems, the yarn or the hybrid becomes strong. We wanted to upscale the textiles, so we had to see which construction technique can be used with thick, soft yarn and with a live paste. And these were our two central questions: which textile construction techniques are suitable for building with MBC filaments, and how can they be evaluated in the context of different architectural applications? For architectural use, the production of the three-dimensional spatial structures is key. So we focused on these three techniques with integrated 3D building potential, knitting, weaving, and knotting. And depending on which construction type we choose, the manufacturing process is also very different. And one learning I can already share is that evaluation criteria, of the for textile techniques in combination with MBC must be very application specific. Like knitting can be very suitable for building pavilions, but it can be too weak for load-bearing structures. So knitting is a mesh loop-based technique. It's very elastic and has a high flexibility, and you need minimum 1 yarn to knit, so it's relatively easy to handle. There are different ways of knitting. We targeted basically the knit, single knit, double knit, and circular knit to create walls, panels, walls, and columns. Here you see the circular knitting process directly on loom or on the scaffold. Knits work with hanging, but the larger the knit is, the higher the weight. So then the mesh deform, reducing the MBC material and weakens the structures. So then you can also use the bottom-up construction. It becomes more effective here. You see double knitting, but also here, depending on the amount of material, several hands are needed to lift the knit to create loops. Then the structures are sealed with foil and put into growing chambers. You've seen that room before. And most of the samples were growing for 5 days and then were drying. And you see some of the grown solidified results, flat and circular knit with different densities. Weaving, by contrast, requires multiple filaments, minimum 3, that are crossed with each other. It's more complex to handle, and it works also on a larger scale. Also, woven structures can be transformed into architectural elements such as surfaces, arcs, shells, and columns. And we tested different techniques from simple plain weave to like basket weaving, like hexagonal weaving. And woven structures also have open and dense grids, and the denser the grid is, the higher the stability, and also the bio-welding effect becomes here important. Since we work with flexible mycelium yarns that are formable first and then solidify, hanging is a method to form the structures by gravity, so the form is optimized ideally by forces. Hanging in general minimizes the contamination and reduces the contact points. And after the growing and solidifying, they can be turned around, and drying, they can be turned around. So this is after the growing. You see on the left-hand side a densely woven arc and also more open shell structures. Macrame knotting is the third technique. It's a bit of a mix of both techniques. You need also 3 yarns that are interwoven but by loops. It's very complex to handle and relatively weak, but therefore the technique allows to embed predetermined breaking points by squeeze out material on certain points. So after breaking the material on these spots, they can result into movable structures. This video should be in a loop, but I think you saw the flexibility. So the sleeves still keep the surface together, although the mycelium break. So we really tested a lot on different parameters, but we didn't do this alone. We also initiated to workshop with architecture students, and we could test a lot and also scaling up the prototypes. We identified 3 main cultivation scenarios. One, like first the bottom-up scenario or bottom-up construction, the hanging cultivation, and a fusion of both, so standing and tensioning. And we selected actually the last one for scaling up structures because we realized it works the best with scaling up. So we just made in October scaled up tests. I will show now 2 objects that we made, one big column for which we first also tensioned strings and started weaving with MBC filament. And you also see that the weight of the mycelium shapes the column. Here you see it in the growing chamber, and it resulted in a very stable and freestanding, or let's say quite stable column. And then we made a dome, also starting by hanging strings. I don't know if it's visible, but we first were hanging strings and then started interweaving with embassy filaments. The growing was also quite successful. And yeah, as the last part, this was fast. Okay, that's actually a pity because I wanted to show you the intensive body work it takes to weave. Okay, and thank you very much. Thank you for your attention, and also thank you for the committee to make it possible to show the work, and also to thank you to University of Stuttgart, Eliza, Biala, and Professor Ostermann that I was able to conduct all of this research together. So yeah, and make objects like this. Thank you.
Thank you very much. You can take a seat in the first row, and it was really inspiring seeing all of these very different presentations. It was also now very satisfying for me to see that you were in fact working with the sausage making machine, because from my own research I've worked with textile and I know how difficult it can be to get a substrate inside of a textile. So I maybe would have one question immediately about your work. What about the speed? Because you said it is very intensive work, and we have a big topic in mycelium projects and is the sterile environment. So I would imagine that you would rather work in a fast pace, or did you have a feeling that you would need to be quick in order for contamination to be avoided, or did you make sure that the space itself was sterile, or how much, and that is also a question to all of the presenters, how much does the sterile environment play a role in your work? Because as you, Natalia, mentioned, some of your materials weren't sterilized, and so maybe you can afterwards also talk about that.
So we were working in a lab, which of course helps because it's not 100% sterile, I would say, but there's already very good precondition to do these experiments. And what but Eliza is more of the expert in this topic, but I know that we work with a very, very strong mycelium, which also helps a lot, like fighting the contamination. Yeah, for me, my experience is that it's also a little bit of luck. Sometimes you cannot really tell why did it work. I was starting with my experiments working in a very sterile environment, and it worked. Later, I got contamination. So yeah, that's maybe what I can say.
Yeah, maybe I can just add. So the contamination is always part of that. Even if it's not shown on the photos. And I think it's just important to talk about it at some point. I mean, with some of our tests, we had a lot of troubles with contamination. I mean, so they were kind of intended as just testing grounds to really seeing what happens if you really don't sterilize it. So if you just wash it with warm water, with boiled water. And finally, sometimes it works. And, you know, speaking from an architecture point of view, it's important to kind of push for non-sterilizing. So in our lab, we currently don't have an autoclave, which pushes us to certain limits, and it kind of makes us think of, okay, how we make it work without the sterilization. So it's also about this strength of the culture, when is a good moment to put it so we can actually sustain the strength and not be kind of over-competed with some other organism. So it's a bit of an interplay. It's probably nice to have this challenge, or not just relying on the pure sterility, because that also has some other impacts in the environment. And of course, mycelium is not it's not in nature. It always functions as a part of a bigger picture. So.
Exactly. Thank you. We can start taking questions from the audience. I have enough questions, which I will ask later.
I would like to ask the engineer if you have tried to bond mycelium to mycelium with mycelium.
So I, my time is limited. I haven't, but there was actually a very interesting paper that came out a little while ago, looking at biowelding and using mycelium to bond mycelium composite with mycelium composites. And I think, if I'm not mistaken, the bond that they get there is actually stronger than the composite outside. So mycelium is stronger than itself, I guess. Yeah.
Thanks.
So maybe I can follow up. It's again a question to the engineer. This is because I'm a material scientist, so it comes naturally. I would be interested in the potential that you see of these mycelium-based materials or composites for the building industry as compared to other more traditional approaches, like just making buildings out of wood, which is a natural material by itself, or by possibly replacing cement, which is the major CO2 producer, by other more mildly CO2 emitting substances like geopolymer or so. Could you tell us about your perspective, the potential advantages or so of the mycelium-based composites?
So I think to me at least, the biggest advantage I think we have. I think the key advantage I see is the potential for taking agricultural waste and then putting that into the built environment as a carbon sink. Our built industry is very technology adverse. They like to use methods from 100 years ago rather than methods today. So I think the biggest challenge there is acceptance of these methods. With mycelium in particular, I don't think we can replace concrete exactly, but just about everything else in your building that's not supporting large loads, I think, can eventually be replaced by these methods. But I think, in comparison to wood, where we are cutting down trees, that I think can continue, but where it's waste and things that we're going to just let decompose out into the environment, we could maybe take that waste and then reuse it for this. I think that's where the potential lies in the area.
I have also a question for Evgueni. I'm sorry. It's a really small curiosity. Does the metal degrade once the mycelium, because you're hinting that the mycelium might feed on it, so does it degrade when it grows with mycelium?
So that's the backup slides that we kind of went through. But anyway, yes. So yes, we think it does. With some of the metals, we see a lot of degradation. So with zinc and nickel, we see like 1 to 2% material loss, which is substantial. So it is degrade. Something that really bonds well is copper or brass, and we don't see any noticeable. So it must be under 0.1%. That's where our resolution tends to degrade, and we just can't measure it. We're sure it happens. So if we look at SEM images of the copper before and after, you can see there's a little bit of pitting. So basically, it's attacking the metal, but the amount it's using is very small. So I think there's potential for chemical treatment, having copper in a solution. So you're not actually putting in a chunk of copper, but you're actually using copper as a salt, and that could be a good way to strengthen it and improve and get that growth. But yes, we think that we're quite sure it's taking the metal out. Yeah.
And maybe to jump into that question, what about rust? How does rust work with mycelium, or does it?
I'm not a chemist, and maybe the material scientists can jump on this one. With the iron composites, we do see explicit rust, and when we put them in, they're all mild steel or mild iron is already rusting. So we do see that rust enhanced. Basically, it's much more, you get much more rust happening with the mycelium than you do otherwise. The break when you have rust or when you have steel tends to be at the rust, not at the mycelium. Everything else, it tends to be the mycelium that's breaking. So it's actually in just that one specific scenario that we've tested with the iron, the break happens at the rust. So I think there's more because copper oxidizes, and that could be considered rust in some way. So it's chemists would be better equipped to.
I have another question for all of you. What is the next step to jump out of the prototype and lab? So what do we need as a next step to get this into either a product or a usable process on a construction site? What would you think, where is the gap? And maybe you can briefly, all of you can briefly make a statement to this point. That would be interesting. Thank you.
Okay. So I think like what we presented here was more like speculative construction technique. And when we want something to get onto a construction site, we need actually the industrialized product are the method, right? So like we made a proof of concept, which is promising. And now we just have to jump like few more technology readiness levels. So like to have the repeatability and like really understanding all the parameters of the material and the process to be able to like guarantee the like the success on a construction site and being trustworthy for the further users and how it ages. Like there's like a lot of questions we didn't tackle within the process. So I think what comes after the proof of concept is basically like research project with like a longer time span when we can try one after another, try to answer the projects and then to really have the very down to earth scenarios. So time and money in a way.
Time and money.
Yeah, I'm asking myself the same question. I think, yeah, time and money is probably a really good point, but I think there's a lot of testing required, but I think the more the more I work in this field, the more questions I have that need to be evaluated. There's so many, as I try to boil it down to several parameters. It's like amazing how many different parameters you have to deal with working with mycelium, like combining it with different materials, like every temperature change has an impact. Yeah, there's a lot of things to do, I think, before we can move on this step. But I think the more we test, the more we find out, the more we share probably also knowledge, the better we can process and move on.
Maybe I can add that in terms of applying these things into building scales or architecture, usually the problem is also with the certification, with the kind of expensive processes of going through standardization that was mentioned before, the kind of intensive testing, and getting the materials certified so they're safe to be used within the buildings. And depends on the country, but usually they're quite strict. And then, as Evgueni said, that the industry is quite slow in absorbing new techniques. So somehow, something that I think was mentioned as well, we need to kind of prove and attempt these scales to kind of push it to the next step. But at some point, some policies will need to be a little bit updated, and the kind of regulations also on the level of European Union would need to also kind of allow for this new class of materials. And really, the process itself seems extremely lengthy. So we have some time for the research in the meantime. But yeah, it's quite tricky. But it's important to also apply it in smaller scales and kind of delivering products and really kind of pushing it from the social side. Yeah, maybe I can pass it to engineer.
Anything to add?
I think it's all been said. So
thank you very much.
Okay, perfect. So I think we can start. Welcome back after the lunch break. We're now going to have a very interesting topical session, as already mentioned, about bio-inspiration. Maybe just to very briefly introduce myself, my name is Helga Lichtenegger. I work at Boku University. I have a material science background, and I'm very excited to guide you through this session. So, what is bio-inspiration about? Well, we have already heard about many fascinating examples of how to combine biological materials and non-biological materials. In bio-inspiration, it can also go in that direction, but it's a little bit more. It's basically about the concepts that we find in biology. So as you all know, in biology, we have fascinating materials like, for example, wood as an extremely strong, lightweight material, or seashells as extremely scratch-resistant materials. And we also have concepts like this co-creation and this circularity. And bio-inspiration is about taking one or more of these concepts and really translate them to an application or a completely new concept that can be used by humankind. And at this interface between biology and human technology, we're now going to have this session, and it's my pleasure to very briefly introduce the speakers. So we will have Ile Gebeshuber from TU Wien with her talk about her talk is From Plant Galls to Grown Devices Towards Programmable Living Material Inspired by Plant-Insect Interactions. The second one will be given by Florian Zischka, also from the TU Wien. His topic is Butterfly Wing Scales as Inspiration for Multifunctional Architectural Surfaces. The third one will be Ilaria Mazzolini from the EM studio in Milano and Los Angeles. She's also faculty at the Southern California Institute of Architecture in Los Angeles. And her topic is Nature-Inspired Construction and Reconstruction, Biomimetic Solutions for Resilient Buildings, Building Assemblies. And finally, we will have Clemens Preisinger from the University of Applied Arts, and he will introduce the Root Arch Research Project. So I'm already very much looking forward. Okay, so and then I would like to hand over the floor to the speakers, and Ile is the first one. May I ask you to come up and give your presentation? And maybe while Ile is coming up, just a short organizational announcement. We will have the talks first and the questions then jointly afterwards. Okay, so thank you, and Ile, the floor is yours.
Thanks, Camille and Petra, for organizing a wonderful conference. I was on Monday at the Wirtschaftskammer Austria, Chamber of Commerce in Austria, on a conference on a similar theme, and I can tell you it was not as it is here. So I'm happy to be here. I'm going to talk about plant galls. So, what if architecture can grow? What if walls would be possible to heal themselves and disassembled at the end of their life like leaves in autumn? I'm a physicist from TU Wien, from the Institute of Applied Physics, and I am exploring living materials also in the context of architecture and cultivated matter. So what you can see here, and all the photos until the end of this presentation, is plant galls in Yorkshire, in England. I was a couple of months ago at the 40th annual meeting of the British Plant Gall Society, which is a community of about 20 people who are all way above 80 years old. They mainly come from botany, and they are absolute specialists in plant galls and find them by turning leaves around. So we were walking through the forest, we were turning leaves around, and we saw these little miracles. You can see my finger here, so you can imagine how small such a plant gall is. This is a silk button gall. When you move the leaf, it really looks like a golden piece of silk woven into one of these beautiful buttons, which you might know from Chinese clothes. So what is a plant gall? Here you see a cherry gall and some other silk button galls on an oak leaf. A plant gall is a perfect example for interspecies co-design. What happens? Insects or bacteria, and sometimes even flowering plants, but in most cases insect, approach a plant, in most cases oak. We don't know why it's oak, but in most cases oaks are approached, and these insects deposit their eggs, and the eggs are starting to send signals, cues to the plant, chemical, epigenetic, you name it, and then suddenly the plant starts to grow a house for the insect egg which is developing. And it's not just growing a house inside which the insect is living, it's also providing nutrients, water regulation, and so on and so forth. What can we learn for architecture from this? Autonomous fabrication, optimized performance, and locally sourced materials. I also like these two plant galls, again on an oak leaf, and I can tell you, whenever you see a plant gall, you can name two names, if you can, if you're an expert, the name of the insect which induced the functionality and the species of the plant which is growing the plant gall. So it's a very, very, very specific interaction, how the insects are talking with the specific plant and how they are reprogramming the plant so that it provides the structures and nutrients and protection that these insects need. So it's designed through growth, not through assembly. This is another, well, it can be defined as a plant gall depending on how you define plant galls. This is a leaf miner. So you can see on the structure here where the insect was very tiny. Up here, it lives inside the leaf, inside the two layers, and it starts to develop and starts to eat. And while it's growing, it's moving, and it's getting larger and larger, and then it flies out and lays its eggs on another leaf. I think this is very beautiful, and I was photographing a lot of such leaves in Yorkshire because, of course, these paths always look different, and I think they are just amazing. So plant galls are, of course, natural biocomposites. They have inbuilt insulation and armor. They deter against herbivores. They taste, if you try to eat them, like very bitter substances. Some even have timed openings for emergence of the final insect. So it would be an exit hole through which the final insect can flow out. And in rare species of plant galls, I found one in the Cardinal König house in the 13th district. You have ballistic seat-like launches, like a rocket launch. So you have the plant gall on top of a leaf, and then you have a rocket-like thing where the baby inside sits, and then this is ejected and goes into the soil and stays there over winter. And in spring, the insect would come out and fly around and mate with another insect and cause new plant galls in other trees. So interesting that the performance is already integrated into the material. I love this image of these silk button galls. For this, I had to destroy this leaf. I was very sorry about that, but I like it when I fold it and you can see on top this beautiful woven structure and on the bottom the beautiful woven structure. And in each of these little houses is one tiny little insect, unless some others come. The friendly ones would use this house and would co-inhabit the house with the insect which is living in there. The not so friendly ones would put their eggs into the developing larvae so that inside the larvae the next insect develops and eats the growing larva, and this might even, as a hierarchical superstructure, change the appearance of the gall. And then there are further insects where which put their eggs inside the eggs, which are in the developing larvae, and in rose galls, there is even 5 iterations known. So an insect which puts its egg into an egg, which is in an egg, which is in an egg, which is in a developing larva in one of these plant galls. And with each additional level, the appearance of the gall is changing because they are all reprogramming the host organisms. So it's absolutely amazing and fascinating. Questions for our research. Can we humans become gall wasps? Can we induce plants to grow our architecture? What would be goals which we would like to achieve? Structural components, tools, responsive facade elements, living in infrastructure, you name it. You could think it in any direction you want. The photograph I show you here is a rose gall on a wild rose. And remember, this plant gall material is from the genetics, the host plant. So be it this beautiful silk button gall, this is oak genetically. This is not insect genome or something, some mixture. It's oak epigenetically and chemically changed. Similar for this rose gall, which is spiky and a little bit red. Or here, you might have seen these ones. In Austria, they are known as Schlafäpfel, sleeping apples. In medieval times, they were put under the pillow so that you can sleep better. We look as physicists at plant galls because we are interested first in the growth mechanics. So how does something grow in functional shape? Like we heard in the first keynote, with local materials at ambient pressure, at ambient temperature, with water-based chemistry in functional form, and at the end of the lifetime, it can be used as food or fertilizer for other organisms. So we find this absolutely fascinating. We're also interested in the chemical signal propagation. If you remove such a plant gall from the organism, if you remove such a silk button gall, or such a spiky gall, you would see that the attachment is just a couple of cells. So through this tiny entry door, the chemicals come into the plant and reprogram the whole plant. So here we are still far away from understanding how this happens, especially because the botanists say, we don't want to study plant galls because there are insects involved. And the entomologists say we cannot study plant galls because there are plants involved. So it's a very highly interdisciplinary field. And we as physicists thought, well, if nobody wants to study them, we study them. And we look into some of their properties. This year it was very, very wet in England, so single leaves of oaks were invaded by various species. This is called a knopper gall. Here you have the silk button galls. So a conceptual pipeline of our work is to study the gall formation life cycle, to map the functional correlations, and to transfer the principles to living engineered materials. So the new paradigm would be to introduce 3 shifts from traditional to a bio-inspired future, from manufacturing to cultivation, from dead materials to living matter, and from end-of-life waste to circular decomposition. So the vision is to achieve architecture that self-grows, adapts, heals, and returns to soil at the end of its usage. And with this, I come to an end of my presentation and say thank you very much.
So thanks a lot, Ile, for this very inspiring talk. It's my pleasure to now announce our second speaker, Florian Zischke, and he will tell us something about how butterfly wings can be used as an inspiration.
Yeah, hello also from my side. Thank you very much. Unlike Ille, I'm also a physicist, and I studied in her research group, did my bachelor and my master's there, and also published some of my work already. And in this specific research, I dealt with butterflies and how we can get inspired by them for architectural use, for enhancing our buildings, especially with their multifunctionality. So this is a short program of my talk today. First, I want to give you a brief introduction, a motivation, why we should even look at butterflies, what's so nice about them, except for they look nice and fly around and are beautiful, but that's another topic. And then I want to briefly just cut through my methods and my results and come to a brief conclusion in the end. Yeah, we all know pictures like this. Global warming is really damaging our health, damaging our cities. They become wastelands. They overheat. Every year we measure new heat waves, new heat records in urban environments, and they continue to harm us. So why is that relevant in relation to butterflies? Well, butterflies have nanostructures on their scales. You can see one butterfly here. Does it work? Yeah. They have on their scales tiny on their wings. They have scales like this. They are embedded in these pockets. So that's the wing membrane. And on top of the wing membrane, there sit these scales. And on the scales, they have nanostructures, which look like this, or similar to this. And these nanostructures enable the butterfly to have amazing properties. They can range from hydrophobicity, self-cleaning properties, to even structural colors. You may know this morpho butterfly. He is only blue because of the nanostructures on the wing. There's no pigments or no chemistry involved. This is mainly a physical effect. And also there might be passive radiative cooling inside those butterfly nanostructures, which would mean that the butterfly can regulate their temperature just because of the physical structures on the wing. And it would be a nice application to have on our buildings, right? So when we could just structure our building surfaces to have passive radiative cooling properties, so the building can cool itself against the environment and even be cooler than the surrounding temperature. And we wouldn't need air conditioning anymore. So yeah, that was my research question. And how I did it was basically first get a lot of different butterfly specimen. Don't worry, I did not kill anyone or did not harm any butterflies. I collected them all from the butterfly house where they die naturally, and they collected them for me, or from old museum collections that were not needed anymore. And I put them under a thermal camera. And I tried to find differences between species, but also between different parts on the wing, where I find higher emissivities, so parts where the cooling effect would be stronger. Then I looked at those species that were interesting with scanning electron microscopy and compared the nanostructures. Then I tried to reproduce these nanostructures that I found using different materials from a dental office. You might have had this in your mouth at some point in your life when you have an impression from your teeth or something at the dentist. This like duplicating silicon is the name for that, and it's really good at imprinting nanostructures actually. And then I also did some weathering experiments to find out how well these structures actually respond to temperature and to humidity, and over time, how resilient are they, how durable are they, to get a bit of a feeling for that. Yeah, these are my results. Well, the most promising one is this on the top. Here you can see it without irradiation, and then after irradiating it for 15 seconds, you can see these spots here, they really light up, these strips on the forewing, which means there is high emissivity going on in that area, and which could mean there's something good happening for us. So high emissivity means it can cool itself. And there's also some other species show enhanced reflectivity, meaning there's less emissivity in these parts. where it's darker. And that would mean there's lower emissivity and therefore more reflectivity or transparency. I also sent some samples to Milano in Italy, where they have a really nice hyperspectral imaging camera, and they also showed the same results as I had with my thermal imaging, but more exact because they can they can show it over the different wavelengths here. But they found in the white spots, which would correspond to the light blue, lower emissivity over all wavelengths. Structural results were especially interesting because we found a variety of different nanostructures. This is all from the same butterfly, and depending on which scale you look at, the nanostructures look completely different. We would not have expected that, and it really opened up a new field because there now are so many nanostructures, and we don't know what any of them do, and it's really, really hard to find out because nature doesn't come with a script where you can learn about what does what. So that's really a challenge at the moment. Reproducing the nanostructures also showed some promising results, for example, with wax. But as I mentioned, the duplicating silicone is the most promising candidate for that. Here we only had the problem that it crept beneath the scales, and then when we pulled it off, it extracted all of the scales, so they are actually embedded here, which is not what we want, so we have to work on that further. Yeah, and the weathering experiments are also very, very interesting because we could not see anything. There were no changes at all to the not weathered scales. All of the scales were completely undamaged, and in addition to that, the sample we investigated was already over 60 years old. It was from the Museum of Natural History in Vienna, and they had it lying around for 6 decades. And then I put them in the weathering chamber and unleashed all of these different temperatures to these poor scales, but they still were not damaged at all. So we have clearly not reached the borders of durability here. Yeah, to come to my final conclusion, in nature, as I already said, we already have integration instead of how we humans try to build something. We always add a new feature. We pursue additive construction. In nature, it's already integrated in one and the same structure. And in addition to that, especially in butterfly wings, we have a high degree of multifunctionality. There is hydrophobicity, as I said, self-cleaning properties, passive radiative cooling, in some species also color, which all of them would be nice to have in a building, if you ask me. But it makes investigations quite complicating because you can't only focus on one functionality because you only have this structure and nothing else. And you, as I said, you don't know which feature causes the function. And also what I found out in my research, that different species seem to pursue different approaches because we found so many different emissivities across species and especially different structures across the different species. And with that, I would like to thank you for your attention. And if you're further interested in my butterfly research, I made a short video, which is on YouTube and understandable even for non-scientists. And you can watch it with this QR code. Thank you very much.
So, thanks a lot for this very nice presentation to Flora and Zizka. And now we come to the third contribution, the presentation by Ilaria Mazzolini about nature-inspired construction and reconstruction. The floor is yours.
Thank you. Thank you, everyone. Thank you to the organizers for having us here today. Today, I am going to present a project that I'm starting to develop, also in the light of what happened in Los Angeles 11 months ago. We have become familiar with some of these images that highly impacted the city with the fires that occupied 2 large areas, but also throughout the Southern California region, there were a multiplicity of fires. and the burning of 16,000, about 60,000 structures in the neighborhood of Altadena especially, and in Pacific Palisades, together with the disruption of many other living organisms, from large mammals to microorganisms. We are still assessing, after so many months, the disruption caused by these fires due to climate-induced human changes. These are some data, but what is important here is really the toxicity that emerged after the fires. That brought me back to think at materiality, something that is close to the work done in the past years, but again with a new lens in a way, and with the importance of thinking the effect of our human-induced activities also to other organisms. So is designing with other organisms, but also for other organisms in mind. So this research that is it started recently again is really focusing on addressing the apparent conflict in building design between the need for stability and performance to withstand climate change in the imperative of degradability and circularity that leaves no waste. The central inquiry explores how architecture, materials, and assemblies can transition from inert, toxic waste-generating objects to active participants in resilient, regenerative ecosystem, responding to a need for more conventional abstract structuring and contextualization in biodesign. This work is grounded upon 2 decades of my research, both with Architecture Follows Nature and with Nature, Art, and Habitat residency, that in the past 10 years has been looking at some foundational concepts and now are providing some foundations for the hypothesis of coexistent architecture. This interest will found and has founded and continue to found in the next set of research a perspective that looks at edaphology, fields of study that looks at the dynamic interaction between geological substrates, soil, and biological systems over time, to find potential for architecture to be designed with biomaterial responsiveness and slow transformation, allowing to become geobioreactive. But I think the new generations are really interested in these moments of how the architecture can express also the materiality of materials that have a different lifespan, a much shorter, that brings us to the understanding of maintenance and operation in a very different way. Something that we have learned when we look at mud and bamboo houses. This is a small house we did in Ghana, that over time in the past 20 years, we have been patching and caring for in order to coexist with the other living organisms. So it's not anymore to think of the other as a pest, unless it's our pet and we love it, but is thinking of the other as a co-habitant of our structure. And that brings also to a different aesthetic, right? It's a big question for architects, the question of aesthetic, of how we approach repair and operation, and we care for buildings and biomaterials. And in going back to the fascination, the closest or the most recent with edaphology, I'm looking at this idea of inorganic assemblies that can transform and decompose. And by looking at nature and working with other disciplines, we learned that it's always there is a gap between disciplines that we want to bridge, is to understand how soil formation would inform building facades, for example, or some of the elements. So this is where I'm at the moment. So in conclusion, I want to say that together, these lines of inquiry converge in a primary architectural question, one that aligns with the cyclical, evolving logic of nature, embraces transformation over time, and positions architecture in an active participant in climate resilience, moving away from toxicity and extraction towards degradable, circular, and regenerative practices. This early-stage research offers a speculative yet grounded exploration of how architecture can shift from resisting nature to collaborating with it, from designing against time to designing with time. By engaging deeply with processes of decay, soil formation, and environmental responsiveness, this work aims to cultivate strategy for an ecological literate architecture, one grounded in a logic of care and repair, and shaped by material degradability and circularity. Thank you.
So thanks a lot to Ilaria Mazzolini. This was a fascinating insight into perspectives of construction and reconstruction. We now move to our next speaker, Clemens Preisinger. And perfect, looking forward.
Thank you very much for your introduction. It is my honor today to present the outcomes of a research project which was done here at the University of Applied Arts, namely a project called Root Arch, which deals with the potential of roots in an architectural context and how to use principles that can be found in root growth, how these can be applied for architectural structural purposes. The research project is a so-called intra-project which runs for 1 year, which was initiated by Professor Petra Gruber. And we are a team of 6 people doing experimentation and research. The basic idea is to study the growth of roots, to apply digital parametric methods to simulate their growth patterns, and to enable thereof also new production techniques. So this is about it. It's a very much content in for just 1 year, but the project is actually based on another project which was done at the University of Akron, also by Professor Petra Gruber. And there, the potential of roots was, especially of so-called coarse roots, was investigated. Because, I mean, roots, they are multifunctional, which means they provide, for example, stability against loads like wind for the plant, but also provides transportations, pipelines for nutrients, water, and so on. So what we want is to capture this multifunctionality and give it a interpretation which can be used in architectural applications. So here you can see, for example, pile systems and how they could integrate multiple functions, like, for example, retrieving water from the ground or heat, something like this. In Akron, the project focused on coarse roots, and there the problem is that these are very hard to study because soil is not transparent. So one needs to dig out these root thwarts, which can be quite big, as you can see here. Then they need to be cleaned up, the soil needs to be removed. And here in this project, the resultant coarse roots were then digitalized with photogrammetry, and the result of this was then a point cloud. There were actually 10 of these coarse root systems which were investigated from 3 different types of trees to see whether there are any characteristics with respect to the root system. But from these point clouds, it's quite hard to get any information out. So this data, which comprised millions of points, needed to be processed so that the topology could be retrieved, so the axis of the roots. Also data like the dimensions, the diameter of the roots, the surface, and so on. All of this was extracted and then brought in a form which is easier to process. So from the data, the information was retrieved. And this then led to rules where you can investigate how the diameter changes from one root level to the next, or how the branching angles change depending on the type of root. So there's the fine root system and the coarse root system. And so the idea for this current research project was to combine those views. And we developed some conceptual ideas. The problem with the coarse roots is that they take a long time to grow. So for a 1-year research project, that's not possible to see any changes in such a system. So what we did, we concentrated more on the fine root system. And these fine roots, they, depending on the type of plant, they grow quite fast. And so at the starting point of the project, we sat down and tried to think about what to do. And so there were some ideas here. We went along several avenues. So one was to use fine root systems to stabilize soil. Another topic was to see whether fine root systems can be used to connect different pieces together. And on the other hand, for the coarse root system, we further wanted to process the data which was there to get more information out of this. So here, for example, a color print where you can see how the centrality changes. So it's the colors represent distance from the trunk, how this is different in terms of different species. Or what we also did is print the data, so to have 3D models which can be physically studied. And also digital models. So based on the growth principles which were found in the previous project, these grasshopper definitions were developed which can mimic this growth of roots depending on the soil, and here, for example, depending on 3-dimensional obstacles which are in the way. So a very essential part of our research project was the lab that we put up with lighting system, automatic watering system, 3D printer, and so on, so to be able to have a hands-on approach to this research project. Also, students were involved in the 2024 lecture by Petra Gruber. We had different student groups which tried to come up with new innovative ideas, as you can see here. So this is one group which 3D printed patterns in order to be able to control the growth of the roots. Another example here, our 3D printer. And what we set out then was to, in this case here, for example, to study the potential of combining roots growth and 3D printing by using different geometries of different granularity and density to see how like for example here, how the roots interact and solidify the 3D print. Or here another experiment which involved Lego bricks, so where the bricks were used to introduce a pattern in the root growth. Or here another thing which is quite similar to a project which you've already seen. So to have a green sausage which contains soil and is then, so to say, a living sandbag which can be placed in or stacked, and then by activating the root growth, the sand bags, so to say, combine and thereby solidify. Also, another experiment was trying to print with soil, which contains also seeds, and then to make, for example, the layers which are normally always loosely connected, or by the connection is the weak point between the layers, to study how the fine root growth can make this 3D printed parts stronger and better integrated. And this here is an experiment where 3D printed parts were connected simply by root growth. So there are two separate 3D printed structures, and by having the roots grow through these specimen, they connect. And as you can see here in the middle image, we achieved also a really load-bearing structure by it. Or here, also an example where three-dimensional spheres were used to have an environment where the roots grow through and thereby form out of these separate spheres a compact, solid body. Yeah, this is one example, I think a very nice rendering of root growth, which is based on this grasshopper algorithm, which I initially showed. And yeah, the research project started on January 1st this year, so we are nearly finished. And now it's, we have to wrap up the results and to find ways to bring the developments which we did to another research project to the next level. There would be lots of things to further investigate, as you can see here. So let's see what comes out of it. Thank you very much for your attention.
Thanks a lot to Clemens Preisinger. Maybe you just already want to stay up here. I will ask all the speakers to come to the stage with me, so then you can directly answer your questions to the speakers. So, okay, now we have heard very inspiring talks and at the interface between technology and biology. And now, of course, the floor is open for questions. Who would like to start? Okay, I see two hands. Ladies first.
Thank you, Ile, for the really interesting talk. I have never heard about plant galls before. I saw them, but I could never figure out what it really is. So thank you. But I have a question. So because you said it's sort of it's being grown from the plant itself and from the leaf. And have you studied, looked in under the microscope the different structure of the gall in comparison to the leaf? And what's the difference? And is there any kind of interesting learning?
So, especially the German-speaking world has been very active in this field for about 200 years. So if you buy the old books where you can see the old Stiche, what is Stiche in English? I don't know. Etchings, where you can see the old etchings, they have microscopically analyzed this to the detail that you want. But you need to go to the used bookstore to buy the original prints, because if you buy the cheap reprints, you do not resolve these fine details. So it's all there, and it's in German, so it's wonderfully accessible for people here in Austria and Germany, and I think we really have a head start if we want to intensify our studies in this direction. So the morphological investigations have been done, and now we need to, add on current scientific methods and approaches and things like this. Yeah.
Okay, fine. Very long it was thought that it was a replica, then the cells would be cells from flowers or from roots, some other thing that they were reprogrammed to mimic something else, but more recent studies said no, it's not that way. It's really something new. So it's genetically completely the same plant, but the cells are not the cells we find somewhere else. They are slightly different to all places. So they are very specific. The cell itself is something different. Of course, there are cells that are the same, but there are very specific cells you can't find anywhere else in the plant.
So I think there was another question. Yeah, here.
Thank you. Yeah, great talks. My question is for the last, the roots, and I think fascinating that you're able to create an algorithm to follow and find the paths. My question is the next step. Roots, if there isn't a path or if there's a small crack, they can go through and be destructive and can break down whatever it is and push and make their own path and expand into that small hole to make it bigger. Have you thought about trying to incorporate that next aspect of root growth?
That's very interesting. In fact, I know that roots can be very, very destructive. So if you have, for example, a green roof, you need to be very cautious that the roots stay where they should, and you need to add some kind of additional layer to block them. But actually, we didn't look into this potential of destruction. This is a very good idea, I would say. Thank you for this thought. Thank you.
Thank you. I have a question for Florian. I think you still have a lot of questions as well into the research, and it feels like you just really opened Pandora's box on there's so much knowledge in there. Where do I start? As an architect, designer, I'm more interested in how do we scale this up? And do you think these nanostructures could be scaled up, or do we need to stay at this scale? Like, do you have any thoughts on that?
Yeah, thank you for your question, and you hit the nail on the head. I started this research project like I think it was 3 years ago. It was thought to be like a 2-month project or something like that, and it really it bust all the borders. When you ask about scaling up, I see 2 thoughts popping up in my head. First, you can't really scale up the nanostructures themselves because they need to be in that scale to enable the properties they enable. For example, when I showed the scales, you maybe saw that there are always the parallel ridges across the scale, and these are for the hydrophobic properties, because the water pellets, they are in that size and they run off because they are this size apart, and if you put them further apart or narrower, it wouldn't work anymore. And the same is thought to apply for the passive radiative cooling mechanism, because it has to be in the size range of the incoming light to interact with that light wavelength. So when it wants to emit some kind of light, it has to be in that size. So we cannot really scale that in any direction because it has to be nanostructured. The second thing I see, though, is that we could apply bigger areas of the same nanostructures. Like, the butterflies are only very small, typically, and for us, technologically, it's at the moment not possible to apply such small nanostructure to a big building. An engineer would think of, like, nanolithography or etching techniques or something like that, which costs millions of dollars for even a small area, so it's not applicable for buildings at all. One approach Ille suggests is having a paint roller, which would mold the nanostructures from the butterfly, and then you could just roll them on the building, which is easily accessible, not cost intensive at all. So that would be one idea to scale that up, I would say. Thank you.
Okay, so maybe I can ask a small question to Ilaria. I was super intrigued by the term microbiome of the house. And I wanted to ask, so do you have an idea or perspective about how to use that perhaps in a controlled or an intended way? At the moment, it seems like this is something that happens and maybe we can accept it better, but perhaps there are also possibilities to use it on purpose to improve the living.
Well, it's on, right? I don't know. The speculation in this project was the microbiome was also because it was referring to our biome. But the speculation in the house was that different microbes or bacteria were used in the different activities. So some of the fluorescent, for example, for the lighting, that there are some smaller scale, but scalability is the issue in every one, as you well explained. So that is one, or there is a bacillum that is used also in fabric for sport, that they react and they open and close. So we had envisioned a ventilation system, or one that reacts to magnetic field, and so it could use to avoid evaporation of the little pool. So it's not very far. I mean, we worked with a biologist on this, as in all projects. So it's not very far, but the scalability, that's where is a bio-inspired rather than, you know, a product on the market, but the inspiration jumps in scales until we don't find the metamaterials that work at different scales that we are working, not me, but others are working on.
So thank you. Yeah, a lot of possibilities here. And as we have heard, scaling is a big topic. Always in biomimetics, it's actually a topic that already bothered Leonardo da Vinci when he tried to build flying ships that were so large that then the Reynolds number became a different one. So this is certainly a topic that is going to pose challenges for us in the future. Okay, it seems like we are completing the session on time. I would like to cordially thank our speakers for this absolutely wonderful session and to you for the lively discussion. And we're now going to the coffee break.
So welcome back to this session titled Artistic Approaches. My name is Ebba Francien-Valdin and I'm head of the textile department here at the Angewandte. And I'm also a bit hoarse today. I hope you can understand me and I hope that my voice will hold. I'm very happy to moderate this session today, and I also want to thank Camille and Petra especially and everyone for organizing this conference and bringing us together. So this session looks at how artistic research and artistic experimentation can shift the way we perceive and engage with materials and our surroundings. Artistic practices produce knowledge that is experiential, embodied, and situated, and it emerges through processes of making and imagining and becomes visible in the methods and modes of inquiry themselves. In the 4 presentations that we'll hear today, you'll encounter textile-based, digital, ludic, and speculative approaches. And each contribution offers a different way of thinking about materials and showing how they can take on an active role, whether as collaborators in a process, agents of transformation, or as catalysts for dialogue and discourse. We will begin with Antonia Ablas, a textile designer and artist who is interested in human-nature relationships and how to reweave nature into the built environment. In her presentation, What Roles Can Textiles Play in Greening Architecture?, She explores how woven and knitted structures can serve as mediums for plants and allowing them to root directly into textile forms. And Antonia develops small-scale prototypes made from natural fibers, and she shows how textiles can offer new possibilities for vertical greening in urban contexts. Our second speaker is Gabriel Caprielian, Assistant Professor of Architecture at the California Polytechnic State University in San Luis Obispo, and the Director of Design and Innovation for Sustainable Cities program at UC Berkeley. His contribution, Postcards from the Future, Singapore 2125, uses augmented reality to visualize past, present, and speculative future shorelines. Through this artistic architectural lens, he asks how a city like Singapore might rethink its coastal environments in response to sea level rise and ecological loss, and how AR can support place-based knowledge, interdisciplinary collaboration, and public engagement. We then move to Ivan Jakaric and Margarete Jamann from the University of Applied Arts, Vienna, from the Angewandte, and the Department of Experimental Game Cultures. The project, Fermenting the Bioludic Toward a Living Architecture of Symbiotic Play, treats microbial cultures as partners in a playful and experimental artistic research process. By working with fermenting cellulose skins, biofilms, and other living materials, they explore how care, symbiosis, and metamorphosis can reshape our understanding of design and architecture. And then finally, we will hear from Stefanie Rentschler, designer and researcher also from the Angewandte, and her project Slime Mold Crypt brings slime mold into the realm of digital security, transforming the organism into an active participant in the encryption process by tying data protection to the well-being of a living entity, one that must be fed, monitored, and cared for. Her project reframes encryption as a fragile relational and interspecies practice of attention and responsibility. And after the presentations, we will have time for questions and a discussion. But now I'm very happy to welcome Antonia to the stage.
Thank you very much for the nice introduction. Yeah, my name is Antonia and I'm a textile designer and artist. And I study textile design at the University of Applied Science in Imhof. And yeah, I'm looking at the human relationship to the non-human world. And I do this by growing plants on textiles. And at the moment, I'm doing smaller prototypes and sculptures. But I started it with the vision to green cities and make every walls and every lanterns and ceilings green just by textiles. So far, I've greened humans, but I'm still wondering how can we get here? So today I'll talk about my approach, what materials I use, what limitations and advantages this has. I show you some other projects from different people, and then possible future scenarios. So what I have is the textile, and I apply the seeds directly onto the textile, so they will root through the textile. Then there can be a pump that will transport the water upwards into an integrated irrigation system that can be integrated by double-layered textile structures. And then the textile absorbs the water, spreads it across the surface, pulls it up a bit, and it will ultimately run down and keeps the seeds moist. There can be moisture measurement and automation processes integrated, and ultimately it's very well suitable for circular systems because the technical parts can be removed, and the technical parts can be removed and the textile can be composted. So this is kind of the most direct translation I have as a prototype from the visualization. So the plants are growing directly on the textile. There's a microdrip hose integrated as well as a scaffold structure, and then there's the moisture measurement. This is another prototype I made during my bachelor's, where the idea is that the water runs from top to bottom, and then there's the water hose integrated and connected to the top part, and it drips onto the textile. And as you can see, it's really green across the whole top, which means the water really spreads across the surface. This is another prototype I made this year, where I used clay to improve water retention, and the textile here is mainly to protect the clay from being rinsed out, and the wool to protect the clay from direct sunlight. So the materials I use are of natural origin only. Otherwise, I would just create special waste that can't be recycled in any system. Linen and cotton have great water-absorbing qualities, viscose as well, but I mainly concentrate on linen at the moment because it's the most direct to nature and it's most more local. Wool has great, it provides some nutrients and it keeps the water well too, but it doesn't pull up the water that great. So what I like to do is to combine linen and wool. I use various textile techniques, but I mainly do weaving and knitting because they can create those double-layered structures, which means I have the textile and then in parts it can be apart, so the irrigation system can be integrated. And in general, I'd say the thicker the textile is, the better. It just holds more moisture and becomes more resilient. It has certain limitations. One is the plant size, so the plants can't be super big. The water demand is quite high because they're big textile surfaces, especially when the plants are not fully grown. It just dries out quickly. And it needs constant watering because the water is just running down slowly. So as you can see here, the top part is much drier and less green than the bottom part. I've tried to overcome this with a reversed cascade system. So there is the first step, pulling up the water, releasing it into the next step, and then the next step pulls the water up again, and then releases, and so on. And then the other limitation is the lifetime. So I'm not in a closed system. So as you can see here, this is 5 to 6 weeks old, and another 6 weeks later, bacteria and fungi come into play and do their job and eat the textile. So it just has a shorter lifetime, but it basically returns to soil again, which is great. Bringing me to the advantages. So I think growing the plants from the seed instead of using pre-grown plants in greenhouses is super cool because no resources need to be used to transport the plants to the site, and it also creates a very dense surface. It's a short life cycle, but it can be used afterwards. It's also super cool for community engagement. So people can come together, create sculptures, and then they can grow. And textiles themselves are really adaptable and flexible, but also the manufacturing process is really adaptable. No machines need to be produced extra. And it's also low cost compared to other vertical greening systems from my calculations. These are some other projects from different artists. So there's the Hydraulis by Alice Marie Acker, Uzumaki's World by Beth Williams, and Be Grounded by Lara Campos. All of them use natural materials only too, and yeah, do super interesting research as well, so I highly recommend to check that out. These are more on the scientific side. The PhD project by Svenja Koine, where she looked at textiles as plant mediators, and then a research project by Andrea Ehrmann, where she compared different knits and how the plants grow on different knits. So how do we come here? So I don't think that textiles can do all of this, and also they shouldn't, because soil is just such a great ecosystem. But I think they can have some advantages, especially for short-term installations with a long-term use. So what I mean by this is we have for example, construction fence, and the community could come together, and during the time of the construction, the textiles could grow, and once the construction is finished, the textile could be used to revitalize degraded soil. Another example, that's the marketplace in Halle, where I live, and it's not very biophilic, as you can see. So installations could be used to show how much nicer this place could be if there just were more plants. And then it would be also super interesting to integrate the system into the built environment, to collect the rainwater on the roofs and then release it slowly onto the textile so the plants can grow. Just to be complete, these were the references I used, and you can have a look at my website, write me an email, or look at my Instagram for further information. Thank you.
Thank you. Thank you so much, Antonia. Is it on? No. Then I invite Gabrielian for the project from Postcards from the Future.
All right. Well, thank you so much for having me here. It's great to be joining you in Vienna. And I'm going to be sharing actually two projects that are interconnected. So starting with the project that I did in Singapore and a project that I've actually developed based off of that over the last number of months, that is a continuation of this Postcards from the Future project, looking at using augmented reality to consider how cities might adapt to climate change. And it was great to see Antonia's vision of kind of how this material scale can actually become a city scale, because this is a very different scale project that you'll see. And the project seeks to do a number of things. One, reframe the challenges of climate change as an opportunity for design innovation, and redefine the edge condition of shorelines as not a line, but an adaptable shore area. And so I'm also interested in how we might collaborate with nature and landscapes to co-create the environment. And this project in particular is using augmented reality as a platform to engage across disciplines and build place-based knowledge, eventually hoping to inspire collective optimism. So we'll get transported here to Singapore to begin with. Like many tidal cities, Singapore is particularly vulnerable to sea level rise. And this is due in part to the massive land reclamation that has taken place in the country. So 25% of the island nation is built on reclaimed lands, and that makes it particularly vulnerable to rising sea levels. And so the natural soft shore has been constructed and turned into a hard shoreline. Intertidal biodiversity has been replaced with artificial beaches. A relationship with the water, once vital for local communities, has now been replaced with water as spectacle. And human-made so-called super trees have replaced the real super trees, mangrove forests, which now only account for 5% of the original mangrove forests on islands. And in a garden city, nature has been adapted to architecture rather than traditional forms of architecture adapted to nature. So when I came to Singapore a few years ago, the national parks had recently started a rebranding of the country from the garden city to a city in nature. And I thought this was a very interesting reframing to consider how this might be applied to the urban waterfronts. And so I was collaborating with climate change scientists and engineers at the Earth Observatory of Singapore, and then created this tour along the historic shoreline. So you can see on the left is a map from 1843, and you can make out the historic shoreline, and everything on the right is built on reclaimed land. And the eventual project was 24 different augmented reality installations, exploring each of the sites, past, present, and potential future. Now, the project utilizes augmented reality at each installation to visualize the different site information and engage the public on each of the sites. So you can see a couple screenshots here of what the augmented reality looks like and how people are interacting with it at each of the sites to really put people in the place itself. And over the last few years, hundreds of participants have engaged and done the tour. And this is really a diversity of stakeholders. So everything from Eventbrite that brought together the general public to government officials and conferences and so forth. And each of the installations sparked curiosity and conversations. And the ultimate goal was building place-based knowledge and empowering local public participation. Also had a lot of collaborators. So in addition to the climate change scientists and engineers I was working with at the university, there was also local artists like Yip Yu Chong, who's a mural artist. And it was actually one of the inspirations for this project for his painting on the back of one of these temples that depicted the historic shoreline. Now, each of the sites includes historic and present maps to contextualize the site information, and then images from the site's past that depict a historical relationship with the environment. So here you can see the traditional atap house built on stilts above the water. And from that same location in the present day, you can see an engineered canal. Now, this postcard from the future visualizes sea level rise adaptation and cohabitation with tidal ecologies, learning from the past, and then speculates on what somebody might write back 100 years into the future to the present. Postcards at different sites layer past, present, and future, where mangrove forests return to their historic location. Coral reefs thrive alongside seawalls. Walls built to combat sea level rise illustrate a juxtaposition in future responses and highlight vulnerabilities in constructing hard edges. Hybrid engineered and nature-based solutions point to a middle ground where shoreline protection and habitat can coexist, and where historic beaches return to sites that have been covered up by layers of history, where tidal pools and beaches are integrated back into the urban environment. And by embracing water, rising seas, and natural systems can be seen as a collaborator in defining future urban form. So if we go to San Francisco, we'll see many of the same things. The waterfront has been built on reclaimed lands, and in fact, most of the financial district was built on what used to be part of Yerba Buena Cove. And this project was begun as a part of San Francisco's Climate Week, where I co-curated an exhibition at the Urban Center, and we turned the interior into a subtidal habitat with this window display, and something that I created with my architecture class that was doing a studio project on the site where they cut out the different aquatic plants and animals, and then also learned about the habitat themselves. And this was a collaboration between government, nonprofit, and academia that is ongoing. So in San Francisco, I created 8 different augmented reality installations that traverse from the historic shoreline down to the present day waterfront. And this was built off of some other research that had been done previously with the San Francisco Estuary Institute, creating these interactive maps of the historical ecology, and then additional research locating historic photos and information about each of the site's historic past. So if we go to site number 1 on Mission Street, you can see a road is being constructed in 1917 for automobiles. But it was once the location of sand dunes and a coastal prairie. And now it's completely a car-centric concrete street. So perhaps in the future with flying cars, it might be returned back to its coastal dune prairie habitat. Now, Site 2 takes us to what was once the historic shoreline. The street that historically marked high tide is now filled with buildings and driverless Waymo cars. As sea levels rise, water could return with a managed retreat, removing city infrastructure at risk and allowing the bay to reclaim its original habitat. Site 3 takes us to the historic shoreline at Market Street, where ships are still buried beneath the city streets and building foundations. Transforming the street into a canal can be part of a larger adaptation strategy by storing excess rainwater and relieving pressure on rising groundwater and saltwater intrusion caused by rising sea levels. Now, if you were standing at Site 4 in the early 1800s, you would have been knee-deep in water or perhaps mud with the bay fill. And this raises a question: what tidal ecosystems have been covered up by city streets, and could they coexist with future development? Imagine streets as tidal pools. Site 5 envisions Montgomery Street transformed into a 700-meter-long public pool during the summer, and during the winter, when we have our rain, it becomes a reservoir for stormwater. And this is actually a reminder of a 300-meter-long saltwater pool that was actually built 100 years ago in San Francisco along the beach. Now imagine walking in downtown San Francisco through an oak woodland forest. How can that experience change your outlook on the day? Oak trees not only provide shade and shelter for many species, but also provide a significant role in carbon sequestration. So I'll just end here by showing you a quick video of one of these AR installations. Great. Thank you so much.
Thank you so much.
And if you're interested, I have some postcards from the future. Come find me afterwards.
Thank you, Gabriel. Now it's time for Ivan Jakaric and Margarete Jahrmann and their bioludic fermenting project.
Switch to the bioludic. Okay, I'd only take 1 minute from the 12 we have in order to introduce also the speaker of the day. My name is Margarete Jahrmann. I'm an artist and the head of the Experimental Game Cultures department here at the University of Applied Arts. We were founding 3 years ago the Bioludic Lab in the Experimental Game Cultures as a space for experimentation with materiality and where play with materials and materiality and also non-human play finds a space. And the speaker is Ivan Jakaric, who is architect, but also one of my alumni, or our alumni, and was running this lab for 2 to 3 years almost. And I look forward to what the update is, and I'm sure we see beautiful, playful arts. That's what I always see with Ivan. Thank you.
I'm a human, a bioludic artist, and I define myself in the spirit of bioludic as 0 or O negative. You might think now I'm talking about my blood type, and it's true. But for me, it's more about perspective, starting from 0, from the small, from the microscopic, and then growing towards the large and the macro. I truly believe this is the right direction to begin with something that is tiny, fragile, but alive. In the Bioludic lab, we work closely with microorganisms, silent companions that speak without words, but yet create the entire worlds. The artistic practice and research moves along the edge of the visible and the invisible. It goes beyond human senses and between two scales. The microplanetary, where nanoforms of life are too small for us to see, to taste, to hear, and the macroplanetary, where I look at extremely large gigaprocesses that lie far beyond the human perception. The definition of bioludic can be simply expressed through this equation. Moving between nano and gigascale, play becomes an experimental landscape of infinite experience in transformation. It is also conceived both as a portal into limitless world of imaginary solutions, but also as a condition of possibility for world building and its transformative experience. It ferments its own manifesto for all living matters across all scales, guided by non-colonial principles: symbiosis, metamorphosis, interface of care, and sustainability. In this sense, the word bios stands for life. And here I want to quote one of my favorite pieces of graffiti from the time of Solidarność in Poland. Life is a fatal sexually transmitted disease, and the mortality rate is 100%. But there are over 100 definitions of life, and all of them are wrong. In this sense, life resists any single definition. It is relational, always more than human. But however, there are scientists and biologists, and they still want to answer the question, this fundamental one in the biology. What is life, or what is the difference between something that is alive and something that is not alive? Paul Nurse frames this question through 5 great ideas in the biology. The idea of cell, the gene, the evolution by natural selection, the understanding of life in terms of chemistry, and understanding of life in terms of information. But one of my favorite American biologists, Lynn Margulis, in her paper on the origin of mitosing cells, explains that eukaryotic cells evolved actually through this process of endosymbiosis theory, which means that the organelles like mitochondria and the chloroplasts were once free-living organisms, bacteria and cyanobacteria, that were eventually engulfed by other, bigger cells. And all this reminds us that bacteria are not just distant life forms or separate from us. In fact, they are our relatives, shaping our future and present. Ludus stands for play and hosts a method. This is the way of experiencing the body, the excess of body, and the opening towards the possibility of their metamorphosis. In the work in the Bioludic lab, which is redefined as a set of bioludic conditions in which microorganisms and time create new kind of perma-reality. One of the key play mechanics in this work is the fermentation, a true game changer. Put simply, fermentation is microbial metabolism in play, in the action. From a biological point of view, fermentation is catabolic metabolic process in which microorganisms such as bacteria and yeast and fungi actually break down organic substances, usually sugar, and creates new compounds. It is an anaerobic energy-producing process older than oxygen respiration on our planet. And according to Louis Pasteur, fermentation is life without air. But beyond this scientific definition of fermentation, this is also the oldest collaboration between humans and non-humans, an ancient process of metamorphosis. It reminds us that the decomposition and creation are part of the same process. So I don't want to go like deeply in the detail in the fermentation, but there are two types, lactic acid and alcoholic fermentation. But I want to stress that the most important part is the production of molecule NAD+, which actually restart the process of glycolysis and brings energy to the cell. It's all actually about the making energy. And this is also how I want to enter this experience of bioludic playground, beginning with the notion of how we can play within the body to explore the human and non-human body as both host and guest in symbiotic relationship of the fermentation. Through this process, the bioludic artifacts emerge, the bioludic table, the mask, and the food. And here I quote Margarete Yaman. They're not already epistemic objects, but they become such things when they contribute to the momentum of insight. The artistic epistemic object is generated through a spiel, a game, from the idea to object, to discourse, to theory, and finally to findings that allow insights. This is the magic circle of artistic research. And all these bioludic objects are created through the process of symbiotic fermentation. The main protagonist is the symbiotic culture of bacteria and yeast, which produces cellulose-based hydrogel, also known as bacterial cellulose, biofilm, or bioskin. Speaking of skin, our biological skin makes us visible in this world, turns humans into social beings. The bioludic skin challenges these conventional material paradigms by shifting the industrial manufacturing processes to biological growth. It employs living microbial cultures to cultivate materials, embracing fermentation as a mode of making. The bioludic skin appears here as a fourth skin, as a digital and biological hybrid. It is always other than human and symbiotic, a living, playful skin composed of microbial life and digital data. Throughout history, the table as an architectonic object has undergone profound metamorphosis, transforming from a communal public artifact into more intimate one, private object, mediating between surfaces and scales, from ground level to its standard height. The bioludic table is a bacterial architectonic object, a domesticated artifact elevated from the ground, but still very connected with the body of soil. It hosts the play companions Kumbacter and celebrates the rituals around the table and collecting data. The ludic part of this bioludic table is laying out a ludic sensory playground where the gameplay starts and the eating becomes a playful act. The mask, the cultural artifact, and the bioludic object is a placeholder for a pataplay and the permamimicry of the collective human face. It gives a new mysterious identity upon the players, transforming them into the main character of the gameplay, a bacterial superhero. And last but not least is the bioludic SSF, or symbiotic superfood. Explores a largely overlooked area, the edible potential of bacterial nanofibers as bacterial superfood, a speculative taste, a ritual practice around the bioludic table, ingesting, digesting, smelling, and coexisting with microbial life. It asks how edible living materials can be cultivated, not extracted, but co-grown. Drawing on Donna Haraway's notion of companions from cum panis with bread, these microbial companions, AAB and LAB bacteria, are transformed into bacterial jelly, a symbiotic snack, and invite visitors with bacteria, Kumbacter, together around this table. Experimental research here understands fermentation not only as method of food preservation, but as medium of storytelling, a non-colonial ritual of care, symbiosis, metamorphosis, and empathy between humans and non-humans, especially here, bacteria, perhaps because we still carry in our genes the trauma of ancient plagues and epidemics, and mostly perceive bacteria as foreign bodies, as pathogens that must be eliminated. But at the end, in this speculative fermentation lab and kitchen, During the gameplay, visitors ingest bacterial food, inviting bacterial agents to continue their journey inside within the human body. Here, digestion becomes a ludic act of co-creation and cultivation, and transformative experience extended through augmented reality and the gameplay. Thank you.
Thank you very much, Ivan and Margareta. And now, please welcome Stefanie Rentschler, also from the Angewandte, with her project Slime Mold Crypt.
Thank you for the nice introduction. So yes, I'm Stephanie, and I'm a student at the Department of Design Investigations. And today I will be talking about my recent pre-diploma project called Slime Mold Crypt, and also about the process behind it. So at the beginning of the project, I was really interested in mycelium, and especially the growth patterns of mycelium. But in the same time, I was also obsessed with transforming this really haptic thing into a digital pattern. Since we today learned that we share semi-successful ideas as well, at this point I got stuck and reinvented the whole project. And then I ended up at the topic of encryption. So encryption, by definition, is the process of transforming readable plain text into unreadable ciphertext to mask sensitive information from unauthorized users. So basically, every mail that you send is encrypted. And what are the two most used encryption systems today? So the first one is called RSA, and it's based on the factorization of large numbers into their prime factors. And the second one is called AS, and this one is based on kind of a column and row shifting system. And both of those systems can be kind of calculated backwards to a certain point, but you could also just try numbers out if you don't have the correct key. You could say like, is this number combination the right one? Yes or no. So, but since trying numbers out takes a lot of time, and in this case we're speaking about several thousand years, this is nowhere near the relation of human lifetime. So one of the main security indicators here is time. And now we have a new opponent, which is quantum computers. So little introduction of how quantum computers work. Thank you. So classical computers calculate with bits. And each bit could either be a 0 or a 1. And then it has the zeros, it has the ones, it calculates, there's one result. But now quantum computers calculate with something called qubits. And a qubit can be 0 and 1 at the same time. So it can have multiple states, and it also operates with something called the superposition, so the spatial orientation of those little things inside the computer. Fast forward, the result is that quantum computers are super fast, extremely fast. And by then, this time safety indicator of our recent encryption systems is eliminated. So, or at least very much weakened. But what's happening now is that people already start to harvest or store encrypted data because they know that once they have access to a quantum computer, they can decrypt it in no time by, for example, just trying out the numbers. And at this point of the project, I was stuck again because I'm not a computer scientist. I'm a designer who's very interested in these topics. And also I felt a bit lost because I was like, is the only way that my data is safely encrypted that I trust big tech companies? And also I tried to find out lots of things about this topic, but it was a fairly intransparent process at some point. So this confusion formed my key question of what would encryption look like if it were built on personal care rather than depending on the responsibility of tech companies. So now I had this question, but how do we answer it? I got back to the beginning of my project, which was me being inspired by biological organisms. And in this case, I didn't choose mycelium, I chose Physarum polycephalum, which is a slime mold growing in the local woods. And it's just one cell. It only has one cell membrane. So to me, this one cell was the complete opposite of those highly mathematical complex systems. And the way that this slime mold operates is that it spreads in every direction. and then strengthens the little paths that lead to nutrients and weakens the rest. And by this system, it's able to operate its way through nature. And then I did further research and found out 3 major points of how a quantum computer safe encryption system would look like if it would have the same characteristics as this slime mold. So first of all, it grows chaotically. You could never fully predict how nature grows. This could be translated into this encryption system having no real mathematical system behind it, which would eliminate this calculating things backwards point. Then it also operates in decentralized paths. So it's like a knot-like network, and if you attack one point, there's still other ones to operate from. And then the one point that it was most interesting to me is that it does adaptive rerouting. So it reacts immediately to environmental changes. And by knowing that, I did a lot of tryouts. I changed things, saw how the slime mold reacts to it, which is super nice because you can see it under a microscope very well. And a good reaction in this case would be that the little intercellular molecules are flowing fast, which you can actually see with your eyes. And a bad reaction would be that it just completely stops moving. And with those tryouts, I found out that the 3 main points of influence from the slime mold are hue, brightness, and humidity. So you could use those 3 points to manipulate the growth. And in the same time, I also got interested in physical computing and coding. And then it was just like a bunch of information. I had the slime mold, I had the physical computing part, and tried to combine all of this in one project. And in the end, it was this installation. It looks maybe confusing at the beginning, but in the following scenario, big tech companies are no longer responsible for the encryption of your data. But you as a person are responsible for your own data encryption and are responsible to keep your data secure. And for the encryption process, you're using this slime mold that is in the Petri dish. It's very small, so you actually can't see it right now, but it's there. And it acts like a live Tamagotchi, so you have those 3 points of hue, brightness, and humidity, referenced with those 3 knobs. And then you need to take care of your slime mold. And maybe a question that all of you can answer for yourself is, how would the relationship between you and your data change if you are the person responsible for it? You can't just go on vacation because you need to take care of your slime mold, which sounds a bit abstract, but yeah, it is my way of translating this key question into a project. And that's it with the introduction of my project. I hope I could share how you could use biological organisms as an inspiration for also rather speculative projects.
Amazing. Thank you so much. Now I will invite all the speakers to the stage, and there's time for some questions. So thank you all for these very inspiring talks. Thank you so much. I'm just immediately going to ask the audience if there are any questions. We have one here in the front.
Thank you. Very nice talk about the slime mold. Question, how did you transfer the slime mold into numbers for crypting?
So you could work with the pattern, and then it was very much at the beginning, or it was my first slide, that you can use several factors of the optical input of this pattern that's created, and that's changing with the intercellular molecules transforming. And then you can, for example, use what's the average hue of this pixel, can translate this into a number, and then go from pixel to pixel. So every time this optical input changes, the number changes as well.
Okay, thank you.
I have another small question concerning the slime mold and the encryption. So maybe I missed it, but apparently you can encrypt it in a safe way, right? Because I mean, there are these parameters that only you know, and then also the chaotic behavior. But typically encryption is there to send messages to someone else who is then also able to decipher it, provided this person has the right information. So how to do that?
Yes, exactly. It's about this communication part of if person A sends something to person B, it should be a direct way of communicating, and another person should not be able to interfere this process without knowing the correct key.
Sure, but person B is going to decipher it how?
Again, this is a very, it's a speculative approach on the topic. So I don't also know if it completely works in practical matter, because again, but there is something called fuzzy matching. And so quantum computers can answer the question of, does it match, yes or no? And then it's a bit difficult in the way of not answering things as a clear yes and no, but it can also match a certain percentage. And if the other person also has this certain percentage of matching of the optical input, then it could be decrypted.
Okay, yeah, thank you.
Another question right next.
Yes, I have a question to the gaming specialist. I once talked to Professor Barth from Vienna, who was called the Spider Barth because he had many, many spiders in his cellar at Vienna University. And he told me every single spider has its own character, and some of them are very playful. So in the tree or web of life, would you find playfulness in smaller organisms than spiders? Explorative, playful behavior?
Yes, definitely. This is why I'm like working with the microorganisms, especially with the process of fermentation. And there is like maybe, I just want to stress this, that I got like during my exhibition at Design Week, very nice comment. And from the lady, and she told me like, you know, once when you realize that fermentation is everywhere, so you can find it like also in the small pieces around you, on your skin, in the piece of meat, on the orange skin, it's everywhere, inside the body and outside. And it means that the borders between humans and non-humans are very fuzzy. There's no borders actually. So I think like in the terms of the spiders and this playfulness, of course, that is present always, but like if we go like to the nanoscale, it's even more. So imagine that you play with a microscope. So in the terms of fermentation, I was experimenting with yeasts as a fermenting organisms, and it's a beautiful game when you see that around the CO2, it's like a bubble in the water. There are like thousands of colonies of yeasts, and they're playing the game. I mean, of course, you can play also, this is the answer, also on micro level. But also on macro, I want to also to stress, on macro level is also nice to play. Interplanetary and cosmic.
Thank you. I'm really intrigued by seeing those artistic and playful projects. And what also drives my own research is this playing around with stuff that seems to be so undirected and unguided, and sometimes not rigorous enough to call it research. But I'm really interested to live at this intersection of artistic research, playfulness, scientific rigor, and an intention to create something that improves the world. And this is maybe too big of a question, but I would like one of you to comment on it. So this kind of relationship between this rigorousness of science and playfulness, and how we can maybe create another new methodology out of this that helps us further what we're all doing. There seems to be some connections here. Speculation, artistic vision, playfulness, and then still there is a framework of rigorous doing stuff, right?
Maybe I just want to add something to this question. And it's beautiful thing of symbiosis or endosymbiosis between microorganisms. And I'm totally like into this empathy between the organisms, like to facilitate the flat hierarchy between the humans and non-humans. So maybe maybe you can say something.
Sure. Yeah, I think it's been interesting working on this part of the Postcards from the Future project with engineers and scientists who have very different metrics for how they work and like what's successful. So there are things, like we have questionnaires and other parts of the research I didn't share, but it was particularly interesting just to see how different people interacted with the installations. And some people were actually scared to see like these visions of the future, and and then some people thought, oh, this is really interesting and creative. When we did workshops, for instance, with some of the engineers, it was like they couldn't quite understand how to think outside of the box of like a hard engineered solution, and it took basically the whole workshop to start thinking about how you might design with nature and kind of more the nature-based solutions, which they have in another part of the lab, but it's very siloed. So, I think that this idea of playfulness is also maybe a platform for collaboration between people and different disciplinary knowledge, but also collaboration with, I think, nature itself as well. So, yeah.
Thank you. I have a question then that relates a little bit to Petra's question about the process and the methods and the freedom you have if you do an artistic investigation that you also sometimes invent your own methods and processes. And since we've been talking a lot about care and care for living systems, I'm curious how or what role care played in the research process itself, if it did, or how you integrate systems of care or not in the work.
Well, I could take a stab at it. Part of the work with the Postcards from the Future is that, thinking about it could be anything, but also at the same time, instead of just coming up with something for myself, the original idea was that I would actually get a lot of different collaborators that would each create their own postcard. And so there were some collaborators, but in my process, it was mostly looking at the landscape as a collaborator. So all of the future images were based on the past. And so I think that was also the kind of the idea of kind of a care and reciprocity with the historical ecology of that place, and then allowing a framework in the built environment so that it can come back to the place that it once inhabited, and then that also helps our ecosystem as well. So maybe that answers the question.
Definitely, yeah.
It's also a struggle I have been working with, because there are those vertical systems that are just like super automated, and the idea of them is that we as like the end consumers don't need to take care at all. But I think that's not the right approach, because we should be like closer related to the non-human world again, and like how to, like my question is to find this balance between, okay, this works, but it also needs care. So we need to learn to take care of the environment again, and to have a relationship with the plants that grow around us and the food that we have and the bacteria and the fungi. And
yeah, also in preparation for this, I looked at your website, Antonia, and I think you write it very beautifully, because you, at one point, now I can't paraphrase it, but you say something about whenever you have a doubt, the enjoyment or the joy of working with these textiles and the living plants, that's enough of you for knowing why to pursue the work. And I think that's also a form of care, also the playfulness and enjoying what you do. Yeah.
The plants help a lot.
The plants help a lot, yes. Are there any other questions? Maybe we have time for one last question. Okay, then I have another one. I thought Gabriel, you said you would like to inspire collective optimism. And I was wondering if that resonates with all of you. If that's something designers and artists maybe do. Or how do you relate to optimism? Gabriel, you can also expand.
Sure. I think there's a lot of pessimism with climate change, and I think just as a designer, I think we're mostly naturally optimistic, and think that maybe this is actually a catalyst for positive change. So with shorelines in particular, they're mostly not great. I think it's just these hard edges where we've artificially just decided this is where the water is, this is where the land is. And it had a purpose at one point because they're mostly harbors, ports, but usually they're no longer used for that with the containerized shipping. So sea level rise actually presents a challenge that then allows us to redefine that edge condition. And so, I'm always kind of trying to find the positive aspect. So hopefully that creates some optimism with people.
Yeah, I totally agree with you. Personal approach is also, we shouldn't keep reality out of sight, but it doesn't hurt to give a little bit more of optimistic approaches to things in a world that's rather, or the news that we all hear every day are rather negative, I'd say. And try to embrace the things that are positive and draw attention onto them.
You don't all have to comment if you don't want to, but
yes, maybe then the last answer. When I was setting my symbiotic superfood SSF, I think the most important thing was for me, to feel this empathy and to invite the players or the visitors actually to feel it, to take care. And the first thing was to put the humans and non-humans on the same level, on the table. And then when we are all around the table same, I think it can happen, this kind of optimism. And maybe this kind of quantum jump could happen actually eventually in the food design, when we understand the process of fermentation and try to be equal to bacteria in my case, or to other non-humans, or equal with spiders. Why not? It's a ludic act. And it always should be as a ludic act full of joy and playfulness.
Yeah. Thank you. Those are nice closing words.
With joy. What did you say? With joy, we finished with joy and playfulness. I think that's really nice to finish a long day like today. I have the absolute privilege to close this part of the symposium. So first of all, I wanted to thank all of you for making today possible. This includes the speakers, the moderators, but also the audience with questions and your attention. It has been quite a day. Tomorrow we'll start here at 9:30, and then at 10 o'clock we will have the keynote by Mitchell Joachim called Design Against Extinction. So I hope there's a lot of joy and playfulness there as well. And this will be followed by 3 sessions. One is on prototyping and fabrication, one is on bioreceptivity, and we finish with a series of critical reflections. This will be very exciting, I think. And then this will be followed by a short and small aperitif, or small aperitif. I don't know how short it will be. So we will now close the session inside the auditorium, but we welcome you very cordially to join the poster session that is happening outside in the courtyard, the atrium inside here. And we have a series of posters. There's also some exhibits there on display that some are part of the posters and some are part of the presentations that we saw today and as well as tomorrow. And we could all congregate outside, and maybe the authors of the posters also provide some answers to questions if someone has. But I think on behalf of the team, I would like to thank you for this part and see you outside. Thank you.